All right. Thanks, everyone, and welcome back from lunch. We're going to start on time because we have quite a few speakers for this session. And we want to leave enough time for the panel discussion. I know you're all looking forward to that. And also the small group discussion after this. So in this session, we'll be tackling the issue of innate immune response for the innate immunogenicity risk mitigation for process-related impurities. We'll have Andrew Graves, who's going to be talking about, giving an intro and a method overview, validation, control, and sensitivity, specificity, and so forth. And then followed by Jeremy Fry, who will be talking about the ERMI assays for the using the whole blood, some of the critical design attributes. Sofie Denise, who's going to be talking about assay validation as well as using fresh and frozen PBMCs, assay setup such as dose selection and data analysis. Noel Smith will be also talking about some of the assay validations, assay qualification and validations using human PBMC. Then we have Seth Thacker, who's going to be talking about, he's going to give a regulatory some of the regulatory recommendations for assay validation parameters and data analysis. And then last but not least, Daniela Verthelyi is going to, Dr. Daniela Verthelyi is going to talk about many of the things, other additional considerations to be considered for the ERMI assays. So without further ado, I would like to introduce the first speaker, Andrew Graves from the Teva Pharmaceutical.
Thank you, and thank you to the organizers for the invite to share some basic perspective on these innate immunogenicity assays with you today. And as a reminder, a friendly disclaimer that the views expressed are my own and not necessarily those of Teva. So as a quick outline, I'll provide an introduction to ERMIs, introduction to the methods, the controls and conditions, as well as some thoughts on implementation. So to start with, what are ERMIs and why are they important? ERMIs is an acronym for innate immune response modulating impurities. They are process and degradation impurities from the manufacturing process that can induce a response by the innate immune system. Functionally, these impurities can work by triggering toll-like receptors, which in turn trigger release of inflammatory cytokines and chemokines such as IL-6, IL-8, TNF, MIP-1 alpha. And these impurities can be known, which means that they're characterized, identified, and the quantity within the drug product is also known, or they could also be unknown, which means that they have not been identified and they're below the threshold of analytical detection. Previous publications have indicated that there's a risk for innate immunogenicity, specifically presence of IARMIs at low levels were shown to break immune tolerance, which can lead to an increase in anti-drug antibodies and harms the efficacy of the drug. In the finalized guidance for ANDAs in synthetic peptides that was published in May of 2021, the FDA stated that the sponsor should demonstrate that the proposed generic does not contain impurities or contaminants that produce a greater or distinct stimulation of innate immune activity as compared to the RLD. This stance has been reiterated several times. As an example, the draft guidance on the tirzepatide product-specific guidance from August 2023 requires comparative study demonstrating comparable innate immune response risk of the proposed product and the RLD. So while the FDA has not explicitly outlined what tests should be performed, some potential options using reporter cell lines were outlined in HALE, published in 2015, as well as the cytokine release with human PBMCs outlined by Holly in 2021. Whatever testing is used, FDA has indicated that you should plan to test the finished drug product, not just API or individual impurities. You should plan to use 3 lots of your proposed generic on or near release, 3 lots of the proposed generic at the end of the shelf life, and at least 3 lots of RLD prior to expiry. Moving on to the methods. I'm going to start by talking a little bit about reporter cell lines. The use of these reporter cell lines, as I mentioned, was outlined in HALE. Briefly, these are cell lines that express one or more toll-like receptors, and they're cultured in the presence of the proposed drug, the RLD, or controls. These cell lines are typically engineered to express a reporter molecule, such as secreted alkaline phosphatase, and upon induction of the NF-kappa B. And that's a surrogate for cellular activation. Example cell lines include HEC-Blue. These are transformed HEK cell lines that express a single TLR. There are 7 different HEC cell lines available commercially, at least 7, I should say, each engineered with a different TLR. There's raw blue, which is an engineered rodent macrophage cell line. This expresses all TLRs, but at different levels. And they also express NOD2, NOD1, RIG1, MDA5, and Dectin-1. There's also THP1 blue, which is an engineered human monocyte cell line. They are shown to have variable activation with many TLRs, including TLR2, 4, 5, 7, and 8, 8, 9, NOD1, and NOD2. And so how do these assays work? Well, once you have your cells and they've all been placed into the well of your 96 well plate or whatever format you're going to use, you can add your controls, your reference product or drug product to the THP1 blue or raw blue cells. And once they have all made their way into it, you will see over time an induction of response. You see in my example here that we've gotten a response to the RLD, a very similar response to the drug product, and our positive control is inducing quite a robust response. We can then harvest that supernate and measure the alkaline phosphatase activity via absorbance. Once we have the data, we can then perform a comparison. So you can see here some results that we have for raw blue and for THP1. You see that on the left-hand side of each of these graphs, we show drug product in the presence of these example ERMEs, and you see that we see a pretty robust response. Then we compare that to the drug product or RLD on its own, and you see that the proposed drug product and RLD have very similar levels of activation. This would say to me that 1, the assay worked, and 2, that we are likely able to suggest that there is minimal risk between the proposed drug product and the reference product. Benefits to these reporter cell lines is that these cell lines are commercially available. Detection is performed using a common technique like absorbance or luminescence. It can be a relatively affordable solution. The single readout can aid in the interpretation, but there are also some drawbacks. The single readout may not be sufficient. There is no single cell line that typically exhibits reasonable sensitivity to all ERME subtypes, so you may need multiple cell lines. Sensitivity to different types of drugs may vary, and you must control for cell viability at the beginning and end of the assay. So this leads to cytokine release as an alternative method. PBMC cytokine release with the use of peptide drugs was outlined in Holly, published in 2021. Briefly, human peripheral blood mononuclear cells are cultured in the presence of the proposed drug product, the reference product, or controls. And upon exposure, PBMCs recognize the presence of ERMEs, and they're expected to secrete a variety of cytokines in reaction to those ERMEs. So cytokines like IL-6, IL-8, TNF, MIP-1 alpha, and more. This allows for a polyfunctional assessment of innate immunogenicity risk of a product in a single assay. And to visualize what this looks like, we have, again, our 96-well plate. We have our PBMCs, they're thawed. We will count and phenotype those PBMCs and then plate them into each of the wells. And then we will again expose our wells to each of the reference product, the drug product itself, or the various positive controls. After a particular incubation time, we see that cytokines are elicited. And again, for this example, the reference and drug product are very similar. The positive control is causing quite a robust response. We can then harvest the supernate and measure those cytokines. In our lab, we typically lean on a MSD platform in order to assess the various cytokines we're interested in, and we look at 10 at a time. So here you can see how we first analyze to make sure that our assay is working. In this case, on the far left side of this graph, you see the drug product, and then across the bottom, moving to the right, is the drug product in the presence of each of our example IARMIs. And what we're looking for here is that we should see an increase in response when the example ERME is present. So if we see that response, we should see that there is a statistically significant increase. And in this example, all but one have a statistically significant response in the presence of the drug. So this, to me, reaffirms what our sensitivity is of the assay and shows that the assay was able to detect at least 9 of the 10 example ERMIs that we used in this assay. Benefits of this is it's sophisticated and provides a multifunctional readout. It's a better in vitro model of predicted in vivo response, at least in my opinion. It's more likely to detect small differences between products. Drawbacks, it's a much more complicated procedure and much more complicated readout. You may need to perform multiple ELISAs or multiplex MSD or a Luminex type assay, and I would also strongly recommend flow cytometry. The sensitivity can vary by donor, so the assay must be well characterized. Multiple donors are needed, often more donors than what you think. And you must control for the cell viability. We recommend as well that you characterize major cell populations both before and after stimulation. I have seen instances where the stimulant of note caused a wide amount of cell death, and then you see, oh, there's no response. And you may think, oh, that's great, my drug product looks great. Well, if you killed all the cells, then no, it doesn't look great. Fresh or frozen, this is a hot topic for some people. Cryopreserved PBMCs may be much more practical to work with, but the cryopreservation process may impact the sensitivity, so that's something for you to consider when you're developing your assay. Moving on to our controls and conditions. For assay controls, viability is number one. We always want to ensure that the cells that we use in our assay are high quality and have high viability. We monitor the cell viability both pre and post exposure to the drug product, the RLD, and the controls, just to make sure that the final readout has not been skewed by the results of the stimulation. For PBMC assays, we recommend ensuring major immune compartments are represented both before and after the incubation period, so you didn't selectively wipe out, say, your monocytes. That would not be a fun readout for you to try to interpret. We eliminate low-quality samples from the downstream analysis, so if it turns out that, hey, 30% of my cells were wiped out, I have some concern about whether or not that readout is going to be acceptable, I would remove that readout from our statistical analysis. Positive control, we recommend using a mitogen that stimulates a broad response, demonstrate that the cells can be stimulated. Negative control is what amount of activation or cytokine release is attributable just to homeostasis in your cell culture. But we also need to have some type of controls throughout the assay. So one of the things that I recommend, this is what we refer to as a median cytokine control. This is the 10 different cytokines that we are looking at examining in our assay. We have purposely purchased recombinant cytokine. We have cocktailed it at the beginning of the study. We then aliquot it and freeze it, and we pull an aliquot with each of the samples or each of the runs that we do throughout. In this way, we are expecting that we should get a very similar concentration from plate 1 to plate 7, and we can monitor over the run to see how well the assay is performing for each of these cytokines. As you can see from the data I show here, some of the cytokines have very nice tight CV, and other cytokines may not be quite so tight. That can also help you with your interpretation of the data. Regardless of the assay format, suitability controls demonstrating the sensitivity of the assay to various IRMIs in the presence of the drug in question should be employed. ERMI concentration should be at or near the sensitivity levels determined as part of your validation. And in the case of a cytokine release, one or more cytokines should be detectable as statistically significant level, in response to the exposure of the representative ERMI. Again, this table that you're seeing here was pulled from the Holly paper, just as an example for you to go by. For the drug product and RLD, as we've mentioned in the previous slides, 3 lots of proposed drug product at or near release, 3 lots of the proposed drug product near expiry, and 3 lots of the reference listed drug unexpired but as available. Additional controls to consider may be formulation buffer. It may be perhaps necessary to characterize the impact that that buffer and its excipients are having on the assay. Drug substance, If formulation buffer is shown to impact the assay, one alternative approach may be to test the drug substance reconstituted in an immunoinert buffer such as PBS. Note that this does carry some risk, and as a person in industry, I have a little bit of hesitance testing it, and the main reason for that is we don't have access to the reference drug substance. So if you're relatively convinced that your drug substance is not going to be immunogenic, then you're probably fine. If there is some immunogenicity to your drug substance, is that from just the immunogenicity of the peptide itself, or do you have not a good drug substance? You can't really tell because, again, you don't have access to the reference product's drug substance. Moving on to implementation. For validation, to support the submission, plan to have a robust fit-for-purpose assay validation or qualification package to justify your approach and your results. At a minimum, expect to define the assay reproducibility and precision, as well as the sensitivity of the assay to the representative ERMEs that you're employing. Depending on the assay format, also expect to evaluate robustness and stability. For example, if you plan to freeze the PBMC supernatants following incubation and harvest, how long can those supernatants be frozen without impacting the results? For sensitivity, different drugs are supplied at different concentrations. As cell lines and PBMCs may be sensitive to high concentrations of drug product, different dilutions may be necessary for different drugs. In turn, different dilutions will impact the sensitivity and performance of the assay. Here's an example of something that we looked at as a way of kind of confirming what our assay sensitivity and specificity was. In this scenario, we took 5 donors. Each donor had known negative control and known positive control, and then each one was also exposed to 10 different unknowns. The unknowns were mixed by an unblinded person in the lab. They were drug product with ERME, those are the ones in yellow, or they were drug product on its own. The drug product on its own is expected to be negative. The drug product plus ERME is expected to be positive. Once we did this, then the blinded analyst completed the rest of the assay, and upon conclusion of the assay, we compared the results, and you see that when they overlay, that you can spot that there were a handful that had a little bit of a difference, and we are able to then calculate that sensitivity, which is picking up when there's a true positive, was 96%, and specificity, which is picking up when there's a true negative, was 92%. Thought that was pretty good. It's better than most COVID tests. So final considerations and take-home messages. Complex in vitro assays can be a critical component of an ANDA submission, but therefore requires a robust assay validation and qualification package to justify your approach. It is possible that one assay may not be sufficient to determine the comprehensive immunogenicity risk of your candidate product. Rather than relying on a single assay, be prepared to generate a package of data that supports your immunogenicity risk assessment. Ensure that your data package is robust and comprehensive and that you have a sufficient donor pool. You've done repeated checks of cell content and viability. You have a sensitive and thoughtful statistical approach, and have a statistical analysis plan that you follow and is justified rather than choosing a statistical approach that suits your needs. Many times I've encountered where somebody has suggested, oh, maybe we should do this statistical approach instead after we've already gotten the results. And I said, no, no, we pick the statistics before we do it, not after, just to get the result that we want. In cases of multiple readouts, one fair question is, do you analyze each readout individually, or do you need to examine the combined readouts as well? I think that there's still research to be done on that front, so I will abstain from sullying the waters further with my own personal opinion, but just know that that may be a consideration for you. This one I will provide my opinion is that blinded sensitivity specificity check at your assay sensitivity limits is very helpful for you to verify and validate your sensitivity claims. So I would still suggest that as part of your validation. And with that, I'd like to thank especially a lot of people in my group who've done a lot of hard work over the past 3-4 years, and my boss, Michelle, for tolerating me. So with that in mind, thank you for your attention.
Thank you, Andrew. That was a great overview. Next, I would like to welcome Dr. Jeremy Fry, talking about validating the ProStorm ERME assay, insights, and experience with whole blood.
Okay, thank you very much indeed, Eric, for that introduction. So today I'm going to be talking really about fresh whole blood assays. We're here because we think that the ability of the Hermes assays are really critical to understanding the risk of clinical immunogenicity. So innate immunity, as we've heard in some of the great introduction there from Andrew, is really a key driver of some of these risks of clinical immunogenicity. And why is this? And essentially because product and process-related impurities in our drug can potentially interact with pattern recognition receptors. And this can then lead to the activation of innate immune pathways. Why do we really care about that? Well, particularly because if we do have those triggering of innate immune pathways, they can actually break tolerance. And so our drug, or these impurities, have an inadvertent adjuvant effect. And that breaking of tolerance is because you then work in concert with the adaptive immune response that we've been hearing about this morning, leading to this unwanted immune response. So if any generic peptide impurity behaves inadvertently as an adjuvant, then we have this elevated risk of immunogenicity. So what is the best in vitro method to model this risk? Well, in the room, we have a range of opinions. We kind of come down quite strongly on, we think the fresh whole blood assay is a really great model for this for a number of different reasons. But in the interest of non-biasness, I'm going to try and present both the positive and the negative aspects of this. I think the first thing to highlight is that when you're working with fresh whole blood, there's no bias introduced due to cell purification or isolation methods. So if you're working with PBMCs, for example, you're having to go through a Ficol separation process, and that means that the certain cell types are not going to be carried through into that purification to the same degree as if you're just using a fresh whole blood sample. So that means that all cell types in the fresh whole blood are included by default. We have a natural variance of the cell type in ratios as is represented, including cells like neutrophils, which don't really come across well in PBMC assays. We're doing a direct incubation of the test articles with fresh whole blood, so that has a really high clinical relevance. It's as close as you can get to a clinical trial without really doing one. And of course, we can have a good representation of the population with a broad donor cohort, just like we do in PBMC assays. The drawbacks are really that this can be quite an operationally challenging assay to set up. One needs to source donors and perform the experiment in a very short time frame because it's an innate immune response. Because of that, it can also be quite challenging to validate. And tied in with the point that I've just made in the green section at the top there, the assay, of course, is using a fixed volume of blood. So unlike PBMC assays, where you're working with a fixed number of cells, typically, the fixed volume of blood means that this variable density of specific cell types will vary across donors. But that can also be considered as, a positive, just depending a little bit on how you look at it. So how do we perform our ProStorm fresh whole blood assay? The first thing, this is unmanipulated fresh whole blood sourced from healthy volunteers. So these donors are recruited and informed consent is taken on a panel of donors, depending on the size of the study that we're going to be doing. And then typically around about 50 mils of blood is taken into anticoagulant vacutainers and is taken straight into the lab. So within 3 hours, the blood is being incubated with the test material at a range of concentrations for a 24-hour period. Following that incubation, we then harvest the plasma and we quantify cytokines in an immunoassay. I'll talk about that in a moment. And this allows us to do a direct comparison of the ability of the generic peptides versus the reference listed drug to drive any innate immune pathways. So how we validated this assay. So the first thing to highlight is that the donor blood is incubated in triplicate. So each sample is analyzed, is incubated in triplicate for a 24-hour period. We then harvest the plasma. These triplicates are pooled, and then they're stored at -80 degrees for analysis. The analysis is typically carried out in just a day or two after that, because obviously time is of the essence in delivering the data for these assays. But because there is a storage period, and as Andrew alluded to just now, it's important to demonstrate the stability of those samples over the course of the period that you're actually storing them at -80. When it gets to the point of the analysis, those samples are thawed out, and then the plasma is diluted into technical triplicates for analysis. Why are we doing this dilution step? Really, it's to do with the linearity of the signal in the assay that we're using, but I'll explain that in a moment. So the validation steps are required to demonstrate sufficient sensitivity of the assay, drug tolerance, specificity, precision, robustness, accuracy, and of course, suitability of the data to inform any potential immunogenicity risk. So what we do is, first of all, the cytokine panel that we're looking at in the plasma are a 12-point standard curve, first of all, that we generate using individual recombinant cytokines. So this is what we establish to define the measurement of the cytokines in the samples that we're going to be using. So we are looking at IL-2, 4, 6, 8, 10, interferon gamma, and TNF-alpha in our standard panel. And the average cytokine values at the lowest concentration on the standard curve are at least 2 standard deviations above the average signal obtained from our negative PBS incubated test conditions. One thing to bear in mind if you're validating these types of assays is that the wider the panel of cytokines, the more challenging it is for you to be able to do that, because you're going to have acceptance criteria that need to be applied to every single cytokine, and the broader the set, the more challenging that is going to be to pass your acceptance criteria. So the number of donors that you're going to need to recruit for these kinds of studies is going to increase quite considerably. We've performed assay sensitivity analysis, so we've determined that our lower limit of quantification is set at 100 picograms per mil for each cytokine. So based on our standard curve, this was actually determined to be 10 picograms per mil, but because we do this plasma dilution to get the linearity of signal, this has to be taken into consideration, and it's a 1 in 10 dilution, so then the LLOQ goes to 100 picograms per mil. And any data points that are below that LLOQ are reported as 0 picograms per mil in the reports. And then likewise, on the upper limit of quantification, this is set at 100 picograms per mil for each cytokine condition as well. And any data points above that are reported as greater than 100,000 picograms per mil. So assay precision is a really important control that you need to consider. And we do this by doing a spiked pooled human plasma sample. So 6 replicates of diluted plasma that have been spiked with a fixed single concentration of pooled recombinant cytokines is applied on every single plate that we analyze. And this is because we need to demonstrate that the signal that we're measuring in all the assay plates are recovered within the acceptable limits. And we've set those limits to be between 75% of the expected value and 125% of the expected value. So on this graph, what you can see are the range of different cytokines that we're analyzing, IL-2 through to TNF-alpha on the bottom right. And there are vertical red lines. I don't know if you can see the red particularly, these 2 vertical bars. So the left-hand bar demonstrates the lower limit of acceptance, the 75% of signal, and the right-hand bar is the upper limit of acceptance at 125% of recovery. The filled-in diamonds are the donors that are greater than 100%, and the empty diamonds are those that are less than 100%. And this is just one of our validation donor sets, donor set C, which is representative and shows that all the donors analyzed fall within that acceptance criteria of between 75 and 125% of recovery. So again, just to emphasize that these recovery samples must be run on every single test plate to demonstrate the confidence that you have that the signal is being recovered appropriately. So we have both donor acceptance criteria and also exclusion criteria, and these can be actually split into 2 different parts, both donor-related criteria and analytical criteria. So the donor-related criteria relates to the condition of the donors that we're using, and we're using essentially self-certifying healthy donors that are asymptomatic. They haven't taken any steroidal or non-steroidal anti-inflammatories for either 3 or 7 days prior to donation. So steroidal anti-inflammatories for a week and non-steroidal for 3 days prior to donation. And the bloods that come from the donation have to be incubated with that test article within 3 hours of blood draw. The analytical criteria are also very important. So we have to ensure that the negative control, which is our PBS-treated blood, the endogenous responses and the measurement of the cytokine there are within acceptable limits. So if, for example, you come across a donor which has a screaming high response, that might be interesting from a point of view of this donor might be going for an active infection or something like that, but we still have to set those limits and say, no, we're going to only accept donors below a certain threshold of for the individual cytokines for what that is. We also obviously have to ensure that the spiked plasma recovery sample is within limits, which I've just described, and of course the standard curve performance is within limits as well. We've performed inter and intra-assay variability, and this was carried out on 4 replicate donor sets, in 2 independent experiments, which are kind of just outlined at the top of the slide there. And we performed repeat analyses on independent days as well. The positive control samples were used for this particular data set for the variability. And this is our positive control, so not suitability control, but the assay overriding assay positive control, SEB, which is staphylococcal enterotoxin B. And this is a really strong control just to induce cytokines in all the donors. So these plasma samples taken from the blood where the SEB was incubated with those donors were used for the assay validation for this data set. And 4 donors per set, for which the average concentration of the triplicates was within the quantifiable range for all the cytokines was selected, and we demonstrated an intra-assay variability of 12.8% with an inter-assay variability CV value of 9.1 with an overall average CV of just under 15%. So onto the suitability controls, the ERMIs. This is really the critical point to demonstrate assay sensitivity, that you can measure trace levels of TLR agonists, of these impurities that can trigger innate immune pathways. We selected the range of ERMIs in our current study based on the Halley paper, which we've already heard about. The specific concentration ranges and the specific ERMIs were from that paper, but we've actually optimized them, and again, in accordance with the performance of the assay to ensure that the suitability is in line with the sensitivities that we're observing. So the 5 ERMIs that we're using are LPS, FSL1, CLO75, Zymazan, and MDP, which all trigger different toll-like receptors, which are listed here. And each of them has their own concentration range that we run at 4 independent concentrations. When we look across the different donor sets, I'm just going to show you the negative and positive controls here. So this is just looking at IL-6, just as an indication. And we have, whoops, how do I go backwards? Can somebody, red button. Thank you. Right. The 4 donor sets are shown here. So this is our negative control PBS. And then we have our positive control SEB. And you could just see the consistent performance across these 4 different donor sets of the positive control and the negative control. If we now look at just 2 of the ERMIs, so this is LPS on the left and FSL1 on the right, we have a 4 concentration range. So LPS is run at 1, 0.25, 0.05, and 0.01 nanograms per mil. And these are the 4 different donor sets. And this titration effect is very clear to see. And the horizontal median value is shown here in red for all the different donors that are being analyzed. So this allows you to then run this titration to determine what that lowest level of sensitivity can be obtained for each of the different ERMIs measured for each of the different cytokines. If I put this all together, we can now look at a single donor set. This is donor set B for all 5 different ERMIs. And we've got the negative control here, PBS. We've got the positive control, SEB. And this dotted red line at the top is our upper limit of quantification at 100,000 picograms per mil, with a lower limit at 100 picograms per mil. And you can see here this nice titration effect going down. You can see that not necessarily all ERMIs have a titration effect in all cytokines. So here, for example, MDP, you can see that this actually doesn't titrate because you're having to use the same concentration of ERMIs, same titration range in the single incubations, which are then measuring all the different cytokines. The other thing to point out is this lower limit of quantification here, the 100. So for every report that we do, we generate an assay-specific sensitivity level, which is defined as the lowest concentration of ERMI, where the median cytokine response is greater than the lower limit of quantification. So for example, if we look at FSL1 here, the lowest concentration range of FSL1 that we can achieve above the LLOQ is 0.5 nanograms per mil, and that's lost at a lower concentration range. So these tables are kind of really important as a summary to sort of show the limit of sensitivity that can be obtained for each assay and for each cytokine as bits being measured as well. So we report this for each cytokine, not just for the ERMI alone, which is what you're looking at here, but importantly, the ERMI also in the presence of the generic peptide drug product as well. We need to measure cell viability of antigen-presenting cell subsets. That's really important because considering the potential impact of impurities on TLR activation on the different APC subsets, as well as measuring the cytokine production, is really, really important. And we look at a panel of different APC types. So we're looking at B cells, monocytes, dendritic cells, and neutrophils, all by flow cytometry. And this is combined with a live dead viability stain in the flow so that we can be sure that the different cell subtypes are not affected by the presence of the drug product. So here, this is just an indicative panel of data by flow cytometry looking at the dendritic cell subset. So we're gating on the CD45 versus side scatter, highlighting the CD3 negative, CD56 negative, CD20 negative population, class 2 positive, HLA-DR positive, and then the viability stain as well. So you're able just to confirm in the presence or absence of the drug what the effect is on the viability of those individual APC subsets. So on to the very final part, it's really important to think about how all this information then plays into the study design. So for our whole blood assays, we carry out a two-phase study strategy. The first is a pilot study, typically carried out on 10 donors. And the objective of this is to identify the highest concentration of generic peptide that can be deployed in the assay without affecting APC viability. So we do this at typically 4 concentrations, 1 in 5 up down to 1 in 50 dilution. We have our assay negative control, PBS, and the assay positive control, SEB. There are no ERMIs in this particular part of the study. And then we run a single batch of drug product and formulation buffer only, so looking at any excipient effects. And we characterize the cytokine production and then also the specific APC viability by flow cytometry. That allows us to then identify the highest concentration of drug that you can run in the assay ready for the pivotal study, which is carried out on typically a 30-donor panel. And this is really the core objective, is to then compare any innate immune response to the RLD with multiple batches of the generic peptide at the predetermined highest concentration determined from the pilot study that doesn't affect APC viability. We've got the same controls as before, 3 RLD batches, very important, 3 generic drug product batches near release, and where possible, 3 generic drug product batches near expiry, as well as the formulation buffer only. We characterize the cytokine production, but as well as that, we're also looking at the APC viability because, again, it's important just to show that any measurements that you're seeing is because the cells are actually alive and not dead. But this is the assay where we also run the ERMIs as well. So the ERMIs is run alone, but also the ERMIs in the presence of just a single batch of your generic drug product. So that's important to have those two because we can then consider the impact of any change in signal in the presence of the drug product. And then from a technical point of view, it's also really important to understand that you need to spike the ERMIs into the drug product before any dilutions of the drug are performed, because you don't want to have any effects of the dilution then coming through into the assay. So do your spiking ahead of that. So just to summarize, the fresh whole blood cytokine release assay has been successfully validated by Proimmune for generic peptide innate immunogenicity risk. Our methodology has been submitted to FDA via a DMF filing. So that does mean that our methodology can be reviewed by FDA as part of your ANDA submission. The critical design attributes include demonstrating assay sensitivity using ERMI alone, as well as ERMI in the presence of the drug. You need to demonstrate the viability of different antigen-presenting cell subsets, and we're doing that by flow cytometry. And our assays really feature the fastest turnaround times in the industry, just 6 weeks, which include the donor recruitment step. And we work really closely with our clients, the sponsors of the drugs, to support regulatory success, and that's really demonstrated by the outcomes of our customers. So thank you very much for your time.
Thank you, Jeremy, for the insightful talk. Next, I'd like to welcome Sofie Denise talking about the ARMY assay validation experience.
Okay, thank you and good afternoon, everyone. I will be first introducing the assay, which is a PBMC assay, and has been already mentioned several times today. With a PBMC assay, the topic of fresh versus frozen PBMC is a very relevant question. So I will also show some data about that. After that, I will just briefly go over the design and the results of our assay qualification. after which I will discuss the standard setup of a project, how we run it at our facility, and then also to go deeper into how we analyze the data that we collect. So we start from isolated PBMCs that are incubated with test product. After the incubation period, we harvested the supernatant to quantify the cytokine concentrations via multiplex, and then we also harvest the cells to be able to look at viability by flow cytometry. We have qualified the assay with fresh PBMC, but because of the, I would say, high practical advantages of using cryopreserved PBMCs, we also did a comparison of fresh versus cryopreserved PBMCs. So we compared the results of fresh and cryopreserved PBMC of the same set of donors. We could not find any, we could not find any negative effect of cryopreservation, so we did not see any reduction in response to Hermes controls. Of course, the most important advantage is that working with cryopreserved PBMCs allows for a much more time and cost-efficient evaluation of the products. It also allows to perform a selection of donors upfront if that is required. One important thing to note, and it's very important to guarantee that there is no influence on the sensitivity, is that the PBMCs are extensively QC'd before use after cryopreservation. So after cryopreservation, there is one vial, one random vial per donor that is always thawed before use, and then we check the viability and immunophenotyping, but also we test the functionality. So for each donor that is in our PBMC bank, we test the response to both polyclonal and specific stimulation. For the design, we followed for a large part the conditions that are also included in the Holly paper. That is already referenced quite some time today. So we used the same 10 Hermes at the same 4 concentrations. We also included 10 donors. We added some spike recovery and linearity samples. In total, we performed the qualification over 3 independent experiments, and we included 2 operators. The cytokine panel is mostly selected, so it's not completely the same as in the Holly papers, but it's mostly selected on the most common requested cytokines by the sponsors. One thing that we would already like to note, so on the figure there you see the range of concentrations per cytokine that we obtain with this set of Hermes at those concentrations. Within red, the limits that the values that were outside limits of quantification. In blue, the levels within limits of quantification. And you see already here, and we see that in every project, I would say that the levels for IL-4 and IL-2 are always very low. So a lot of these values are below limits of quantification. It's only at the very high concentrations of Hermes that we see some responses there. So the 2 cytokines are very commonly requested, but actually in almost all projects, all values are below limits of quantification, and in the end, they are not included in any quantitative analysis. We looked at this list of assay parameters. So I added in the appendix a table with a short description of the methods, how we evaluated each of these parameters, and also our results. So the slides will be shared, so you can always consult that. But today, in the presentation, I will only focus on the sensitivity and the viability results. So this table shows the lowest detectable concentration in nanograms per ml of each Hermes that we were able to detect. So per Hermes and then per cytokine. And in green, you see the lowest detectable concentration across the cytokines. This is very much in line with the sensitivity that is also described in the Holly paper. So we confirmed that with a PBMC assay, it is possible to detect these low levels of Hermes. For the viability, we wanted to mostly share the observation that we see lower viability for the CD11C and CD14 population compared to the T and B cell population. So this is viability data after the assay that you see for several conditions. And then the color code is donors. And you see that for the CD11C and CD14, the viability is really lower than for the T and B cell population. We think that because these cell types are very sensitive to physical stress, that this may be at least partially related to the harvesting of the cells, and that the viability in the actual assay was higher. We also see this lower viability for these cell types when we see a very good sensitivity, so when the donors showed a good response to all ERMI controls. So in our opinion, this lower viability that we can see for these cell populations are not a reason for concern. If we go to the standard setup of a project, then we do mostly, as was discussed in the talk before us, also a two-stage approach. So we do first a dose selection, which is typically done on 5 to 10 donors, where we only test one batch of RLD and test drug, but then at multiple concentrations. So we do already at this stage the combination with the ERMI. So we test these concentrations of the RLD and the test drug alone, but also in combination with 5 ERMI controls to see, to be able to evaluate the effect on viability, but then also on ERMI-induced response. One comment, or open question actually, that we want to bring today is that we take into account when we assess a concentration range, the maximum 10-fold dilution that is recommended, so that we do not dilute the product too much. But we do see that for some very concentrated RLDs, we get a concentration that is much higher than what we typically use in an in vitro setting. And the question that we have there is if this maximum 10-fold dilution, irrespective of the concentration of the RLD, is always resulting in relevant concentration to test in vitro. After we have selected the dose, we do then the actual comparison of the RLD versus test drug. So for that, we typically use 10 to 20 donors. So I will come back later to the question about the number of donors to include in these assays. There, typically, the 3 batches of RLD and test drugs are evaluated at 1 concentration. And how we analyze this data in this stage will also come later in this presentation. I just added also the table with the controls that we use in the stage 1 in combination, and also in the stage 2, just as the assay controls. So it is 5 low positive control and then 1 high positive control. Before I go deeper into the details of the analysis method, I just wanted, because it's important for later, or it comes back, to first go to the basics. So cytokine concentrations are log normally distributed, meaning that they are not normally distributed on their natural scale, but they approach a normal distribution after logarithmic transformation. It means that it makes most sense to visualize concentration on a logarithmic scale. It also means that if you do a parametric test like an ANOVA or a t-test, it's very important to first apply a logarithmic transformation to the data. And it also means that it is a good way of reporting results is in fold changes versus then, for instance, an untreated condition or test drug versus RLD. Our standard approach and primary analysis is a test per cytokine, where we follow the approach that is described in this paper that you see there, which is an equivalence test. So that consists of actually 2 steps. The first step is the determination of an equivalence margin, so meaning the largest differences that you consider non-relevant. So if you can prove that the difference between your RLD and your test drug is smaller than this equivalence margin, you can conclude that the 2 products are equivalent. So that's the first step. And then in the second step, of course, you have to actually test if the difference between RLD and test drugs remain within these equivalent margins. So this paper is, of course, not specific to ERMI assays. And in the next few slides, I will go more in detail how we implement this strategy for the ERMI assays. So it's a little bit of statistical theory, but I will try to not go too deep into the details. In the paper that I just referenced, you will see the guideline that you can calculate the equivalence margin based on the variation of your data on the RLD data. So for this equivalence margin, you only need the data of the RLD. And then the recommendation is to use an equivalence margin that is 1.5 times standard deviation. So in the paper, you will see that variation between batches should be included, variation within batches should be included. In our assays, we have an extra source of variability, so the between donor variation, which is not described in the paper. What is important is that we do not want to include this source of variability in our equivalence margin. And we sometimes have quite substantially different baseline concentrations between our donors, but that is not a source of variability that we can include in this standard deviation. And that's because it's not relevant. It's not a relevant source of noise in the comparisons of RLD versus test drug. We test both on the same set of donors, so we can do only within donor comparisons. So it's not justifiable that we include this level of variation into our equivalence margin, which will then be unnecessarily wide. So that is important to realize, how we can separate certain factors to be included in the variability and others not is with the mixed model. So that is what you see here, the output of a model, and what you do not want to include in the variance component, you add as a fixed effect. So that's why donor is added as a fixed effect. Those experimental factors that have to be included in the estimate of variability, you include as a random effect. So, as I said before, we have to, if you want to do a parametric test like this, we have to first log transform our concentrations, and then we can run this relatively simple mixed model, and then we can get the variance estimates that includes all the sources that we want to take into account for the equivalence margin. Of course, if we want to go from variance to the standard deviation, we take the square root, and then we get our equivalence margin on a log scale. Of course, it's much more natural for us to think on a natural scale, so we can back transform these limits on a log scale by exponentiating, so you get something that makes sense for us. So in this case, for instance, it's of course simulated data, but here we would say that the products can be considered equivalent as long as the cytokine concentration are not more than twofold, around twofold difference between the RLD and the test drug. Then in the second step, we have to actually test if the difference between the RLD and test drug remains within these equivalence margins. For that, we have to look at the 90% confidence intervals of the difference between the two. So I will not go into detail why it's 90% confidence intervals, but it's definitely not a typo. We can obtain this by a similar model as described before. Because we now have the test and the RLD data, we of course also have to have that added as a predictor. So that is here termed compound. And then we can look at the average difference over the batches between our test drug and the RLD. And if those 90% confidence intervals are entirely within the equivalence margin, we can demonstrate that we have a significant equivalence between the RLD and the test product. So again, here you can back transform the confidence limits to the natural scale. So you see that here we can state with sufficient confidence that the difference between the RLD and the test is between 0.94 and 1.25, which is very well within the equivalence limit. So in this case, we would be able to conclude that the test drug and the RLD are equivalent. Because we do a test per cytokine. We are, of course, multiple testing. So this raises the question if we should apply a correction for multiple testing. We do not do that because the way that we interpret the results. So with multiple testing, you have a risk that a small percentage of your outcomes are false positive. So 5% if you use the default alpha level. And we will never conclude equivalence if only a very small percentage of our cytokines actually show an equivalent result. So that's the reason why we don't need the correction for multiple testing, because we will not interpret individual positive results. If only a very low percentage of your cytokines show equivalence and all the others do not, the end conclusion will be that they are not equivalent. So for that reason, we also don't have to apply a correction for multiple testing. Correction for multiple testing is very important if you want to interpret individual positive results. So if you test 20 compounds and one of those is positive and you want to claim that it's really positive, then you need to have the correction. Here, if you have 20 cytokines and one of those is positive, it will never result in the end conclusion that your two products are equivalent. For that, you want to see equivalence on all separate cytokines. So for that reason, a correction for multiple testing is not needed and is also not appropriate. That being said, a multidimensional analysis is of course an alternative for the testing per cytokine, because of course these cytokines are not independent readouts, so it definitely makes sense to look at some kind of multidimensional analysis. So for instance, a principal component analysis, but there are many others that are relevant and possible, because we do see, of course, a very high correlation between the cytokines. We do that as an additional analysis, but we do not do that as the primary analysis for two reasons. The first is that we get the feedback from sponsors that the results from such a multidimensional analysis are too complex to understand the conclusions. And that the per cytokine tests are much easier to interpret. And also even from a statistical point of view, it's also less clear how we should, which processes to follow to come to a conclusion or an equivalence based on this multidimensional analysis. So I think it's definitely possible, but it's much less straightforward to follow guidelines for that. And then especially also the feedback from the sponsors that is without statistical expertise, sometimes very difficult to understand what exactly is the conclusion. We do that only as a secondary analysis. I listed in this slide some open questions that have some kind of statistical aspect. So the first one is, we all agree, and of course we should do a quantitative analysis, but we do have an assay where we have we can have a lot of values that are outside the limits of quantification. When all values are below the limit of quantification, for instance, for the condition, it's very straightforward how to report that or how to interpret that, but it becomes more complicated when a certain condition have, for some donors, detectable concentration, and for other donors, values below limit of quantification. Because the values that are below the limit of quantification, we cannot really evaluate what the relative differences are in that range. And then for the donors that do have measurable concentration, we can evaluate relative differences. And how we include or not include those donors that have values below limit of quantification can really have a substantial impact on the results and the end conclusions. And there is not really, I would say, a perfect way to deal with that. So we cannot really say that there is one way that does not have any implications. But which way you choose to deal with that will have an impact on the conclusions. So that is one thing I think that could benefit from a harmonized approach. The other question that is very relevant is what is the adequate number of donors to include in these assays? So from a statistical point of view, it definitely makes sense to base this on the interdonor variation that you see, which can be laboratory specific, but we also have often heard of very specific guidelines where people were requested to include, and then depending on source, 10, 20 or 30 donors. So we can easily calculate, based on the interdonor variation, that we see how reproducible the assay is in function of number of donors. So I just put an example here where we can take a low positive control, so where we see on average here, as in the example, a 3.6-fold increase versus the negative control. And then we can calculate, based on the interdonor variation that we observe, how different this estimate is between random subsets of donors, where we of course expect that this average estimate will be more consistent with when we include more donors. But the added value of that really depends on how much variability you see between the donors. And then here I just put the value. So you see that actually already for 10 donors, if you look at the average estimates, 80% of these experiments with different donors would give you estimates between 2.8 to 4.6, so relatively close to the 3.6. And then especially you see that when you go from 20 to 30 donors, you have almost no added benefit anymore, just because the interdonor variation is not so large that there is an added value in including 30 donors. You're already at the plateau of the power curve where you don't gain so much more by adding those 10 extra donors. And as a last slide, I just have the summary. So we have qualified the PBMC assay, where we have confirmed the sensitivity to detect the ERMs at a low concentration. We have shown that cryopreservation of PBMC does not impact the sensitivity, at least in combination with a stringent quality control of those PBMCs, which allows a much more cost and time-efficient evaluation compared to the fresh PBMCs. We typically do a two-stage approach where we first perform a dose selection, after which we test the RLD. And again, I repeat here, the open question that we have is that the maximum 10-fold dilution for very highly concentrated RLD is sometimes difficult to achieve in vitro without a negative impact on the assay. We normally perform an equivalence test per cytokine, where we follow the recommendation of an equivalence margin that is based on the variation observed for the RLD. For us, this works out quite well, so we see that we can obtain equivalence margins that also make scientific sense based on the variation and the sample size that we use. And then, we also pose the question that there should be at least an evidence-based rationale in the required number of donors, because sometimes we get these recommendations that we don't know very well on which they are based. So that was it. So I just want to thank you all for your attention.
Thank you, Sofie, for the excellent talk. Next, I'd like to welcome Dr. Noel Smith to give his take on the ERMI assay validation experience using PBMC. Thanks.
Okay, great. Thank you. Thank you, Eric. And thank you to CRCG and the FDA for the opportunity to speak and contribute to the meeting, as well as be part of the organizing committee here. So I've really enjoyed it, and it's great to see such a well-assembled group of people here in the field. So yeah, I'm going to give you a bit of the same but different as some of the previous talks. So hopefully it's a bit of variety and not too much repetition. Again, we're focused a lot on the PBMC-based assays rather than the whole blood. But I think some of the approaches we take are maybe slightly different to what other people are doing. So I'll start super briefly with an introduction to the ERMIs and why some of them are important. I'll focus on the PBMC assay. So we've done fresh and cryo PBMC, and I'll touch a little bit on both. Touch on what we've done to validate the assay, go through a typical study scope, and then some challenges and discussions for taking into the sessions later. So very briefly, just a one-slide introduction, just to make the point that I think a lot of people mentioned this already, it's important to have lots of different ERMIs that cover lots of different pathways within the cell. It's quite complicated. I'm not going to go into too much detail, but essentially I've just broken them down here into where they're located within the cell. So we think it's quite important to make sure we've got contributors from the membrane compartment, things like LPS, FSL, flagellin, and zymosan all signal through TLRs and other innate receptors on the cell membrane. And then we've got some in the endosome, things like CL075, ODN, and poly-IC. And then finally, MDP is currently the only reasonable one we have in the cytosol. So I think when you select your ERMI panels to go forward into these studies, it's important that you cover at least one from each of these areas. And I think that's an important thing to note. So as I said, we use human PBMC. I think Sofie quite nicely showed the importance of how you collect and cryopreserve your cells, and we certainly agree. It's key that you get either fresh blood or leukopaks collected very quickly and then into the labs for isolation as quickly as possible. So we tend to do this within a few hours of the blood or the leukopaks being taken from the donors. We use density gradient centrifugation to harvest the cells. We've tried pretty much every automated system under the sun, so and none of them work as well as the manual process, but if anyone has any great ideas on how to do this automated, I'd be very much appreciated. Then we count the cells, we can wash them, vial them, and then they're control rate frozen and then stored. And we have quite a complex sample management system to allocate all of the locations of the cells, the number of vials that we have, any QC, HLA typing data that we have as well. And as has been discussed, you can run these sorts of assays with the fresh PBMC or with the cryobank. Everyone running these assays would much prefer to use the cryopreserved PBMCs. I think as Sofie mentioned, it's much easier, it's much more cost effective to do this. You can pre-qualify the donors, but there are downsides to the sensitivity, especially when you've got some of the toxic excipients like phenol that we have in some of the peptide products. So for that reason, lots of these assays have been run with fresh PBMC, which gives you a bit of a headache on logistics. I think Jeremy mentioned that obviously when you get the fresh blood in, you have to use them straight away. If you need to add a couple of hours of PBMC isolation on top of that, that's pushing you even later into the evening, and you have to kind of allow for that. So this is a very high-level schematic of how we approach the assay. This is just showing the example with the fresh blood. So we have the blood coming in within a few hours of being taken from the donors. We isolate the PBMCs immediately, and then they're treated. So we typically have all the plates ready in parallel to the PBMC being isolated. And then just after 24 hours, we can harvest the supernatant for Luminex analysis. And we tend to do around 8 to 13 plex. I'll touch on that a bit later. And then also using high throughput flow cytometry to look at the cell viability, which a number of people have mentioned is obviously very important. We run this assay, everything's run in triplicate, so we do everything in biological triplicate and then take those biological triplicates all the way through the assay. So we don't do any technical replicates, we focus on that biological variation. So the triplicates in the 96-well plates are taken through to the Luminex analysis. So I'll just show you a little bit of data that we've done to sort of validate the assay, similar to what others have done. This is just focusing on the viability. So again, a number of people have said how important the viability is. I certainly agree. So we typically look at this. Oops, sorry, I think I've skipped one. So this is just showing some data with 5 donors, and then we've got the total PBMC, T cells, NKs, DCs, B cells, and monocytes. And then this is just so typically they're above 95 to 99% immediately post-isolation before they're used in the assay. And then the data in the tables is after 24 hours of incubation by flow cytometry. So you can see we received very little drop in the PBMC, T cells, and NK cell viability. I think Sofie showed a very similar data. We tend to see an impact on the APCs as well. It's very donor dependent. And especially with the monocytes and the DCs, I think they probably stick to the tissue culture plastics. They are difficult to harvest without damaging them. So I think we have to bear that in mind when we're looking at the viability of those cells at the end of the assay. And we have to take into account whether they're functional as well. So then if we bring in, obviously this is just untreated cells, just after 24 hours of incubation. If we then bring in treatment with a drug product, or I think this is an RLD in this case, so we've just got 2 columns added for each cell set with a 1 in 10 dilution or 1 in 20 dilution of this particular drug product. And this one does contain phenol. I haven't shown the 1 in 5 data here, but pretty much 50% of the total PBMCs are killed by lower dilutions, anything around about 1 in 5. So we tend to focus on that the magical 1 in 10 dilution that we're trying to hit here and focus on what's the viability after treatment of the 1 in 10. So as you can see, the total PBMC, the T cells and the NKs are not too impacted at the 1 in 10. It goes up even further at the 1 in 20. But the APCs are much more sensitive to the phenol-induced lowering of the viability. Very, again, donor dependent, but we typically see, anything up to 50% of donors being hit lower viabilities after phenol treatment. But obviously, as I said, we have to balance that with, we don't want to over-dilute the product and then minimize or not maximize the exposure to the impurities in the product. So I think in general, with our experience, I think a 1 in 10 dilution, the assay is tolerable with fresh PBMCs, so 1 in 10. The viabilities can be a little bit lower with the cryopreserved PBMCs, and I think that's why there's currently a focus more on the fresh for these sorts of assays. And then moving to the actual functional readout in the assay. So this is, again, I think a number of people have shown very similar slides where we've got 8 different ERMI here. So we've got LPS, CL075, MDP, FSL, ODN, poly-IC, flagellin, and zymosan. And then we've basically got high, medium, and low concentrations. The lowest one is pretty much there or thereabouts with a lower limit of detection in most of the donors. So I think there's a few different ways you can look at this. You can always find really good donors that you can even go 10-fold lower than these in some cases, but that doesn't work for every single donor. So you have to find that balance with what's your lowest limit that works in most of your donors. And this is basically showing the fold induction compared to untreated cells. So this is what we call the stimulation index. And then this is probably easier to see in a table here, where again, we've got those membrane-bound TLR targeting agonists, the endosomal and the cytosol. And this is what typically our lower limits of sensitivity is in the cryo PBMC, and it's very similar in the fresh PBMC, or slightly lower in some donors. I think one of the headaches we have, and I think I've spoken to lots of people in this room about the challenges with LPS, and I think Sofie had 2 different types of LPS that had a very different lower limit of sensitivity, and that's something that we see as well. With all of these ERMIs, actually, you can get batch-to-batch variability, and even vial variability. Even with something like LPS, if you test 2 different vials from exactly the same batch, you can get quite big differences in 10-fold plus in how sensitive your assay is to them, and that's a real challenge in these sorts of assays. So for that reason, we've obviously picked a subset that we use in some of the more pivotal, larger screening assays where we don't necessarily need to take all 8 ERMIs through, and I'll touch on this in a few slides' time. But we tend to focus on FSL and flagellin as the kind of most robust but sensitive membrane-bound targeting molecules, and then CL075 and MDP to cover the endosomal and the cytosolic pathways. So just a couple of key considerations to note and metrics on the assay performance. I think, again, as has been mentioned already, the assay variation is often driven by the donor. Some donors just behave better in vitro. They have different ratios of different immune cell types that are responsive to different TLRs. It's really important to select the right ERMI concentration. I mentioned the batch-to-batch variation. So we actually qualify every batch of ERMIs that we have with that cryopreserved reference donor that we have. So we have a set donor and a set assay that we qualify every individual ERMI batch just to keep an eye on them and before we run them in any studies. We also we found that filtering the supernatants before you use them in the Luminex assay really cleans up some of the data with some of the cytokines, and obviously that's something we checked. What impact does filtering have on the detection of the cytokines? And I'll show you a little bit of data on that. I think somebody mentioned, I think it was Andrew mentioned supernatant stability as well, so that's something that we've done as well to check that in the window that we have those supernatants stored and then to go 1 freeze thaw. Does that have any impact on the detection of the cytokines? And we're also in the process of looking at longer-term storage. So if we freeze thaw those cytokines, run the assay, and then freeze what's left, refreeze what's left, how long can that be stored just in case we need to go back and investigate something further? So that's something that we're in the process of doing. We have an outlier analysis process as well that we run on all of the raw data. So this is just the raw data coming off the Luminex instruments. And we use this to look at the sort of variability across a study. We can have anything up to 100,000 individual data points within a pivotal study. And we pretty much always see just under 1% of outliers being removed by the preset criteria. So that's something we keep an eye on just to check that there's nothing unusual going on in that data set that's been more variation than we're used to seeing. And then also we looked at the intra-assay CVs for these sorts of assays, and we see about 5 to 20% dependent on the cytokine. And obviously it's more challenging to look at the inter-assay CVs with fresh PBMCs. It's something we can do with the cryopreserved, but that's a bit more challenging with the fresh. And we've actually just completed a couple of studies where we've run across our UK and US sites on the same products and the same batches, but they have been either with cryo in the UK or with fresh PBMC in the US, and they were about 18 months apart, and we saw very similar data with different donors, cryo versus fresh PBMC. So that was nice kind of to know that the assay is robust, and we detected very similar responses across those two different studies. And then this is just a quick snapshot of that data I mentioned, where we looked at filtered and unfiltered samples, where we basically just spiked in known amounts of all the cytokines that we typically look at, some of the key cytokines, and also we add in some chemokines to give us a bit more sensitivity to some of the ERMI controls. And as you can see, there's very little difference between the filtered and non-filtered cytokines going through the assay. So I'd just like to move now and just give you a quick overview of how we approach the studies. I think like some people, we do an initial qualification study just to set the conditions before we move into a more pivotal study. We tend to do these in just 5 donors initially. This is just showing an example of a fresh PBMC, a 5-donor study. We tend to focus on just the 1 in 10 dilution. Obviously, we know that going lower than a 1 in 10 if we have phenol is too toxic to the cells. We know that going higher than that is going to get pushback. So we tend to just focus in the 1 in 10, but then look at how sensitive we are in the 1 in 10. So rather than having a fixed ERMI concentration and different dilutions of the product, we keep the product where we want it to be and then see how sensitive we are within that. So we typically just use the DP, or it could be an RLD here, but usually it's the DP. At the 1 in 10 dilution, and then we have 3 concentrations of all the 8 ERMIs alone, and then spiked into the DP, just to know how sensitive we are. We do also check the viability of the cells after treatment with the DP at 1 in 5, 1 in 10, and 1 in 20, just to show that if we go any lower than 1 in 10, that's detrimental to the cells, the cell health. So that usually allows us to select what's the ERMI concentration that we can detect nicely and robustly in the 1 in 10 diluted drug product. If we're testing drug products that we know don't have phenol or anything, any toxic excipients, we do sometimes drop to 1 in 5 dilution. That's probably the minimum dilution we would have in the assay, based on just not wanting to dilute the assay medium too much and generally upsetting the cells. So that's something we can do on a case-by-case basis as well. And then once we've got that data, we can move into the more pivotal study. I think people have already nicely discussed what we need from the DP and RLD batches. So this is the kind of minimum that we need, 3 RLDs and 3 DPs. That's the main focus of the comparison. But obviously we need the right controls. We typically have a couple of concentrations of at least the 4 key ERMIs that we run here. Sometimes we run all 8 ERMIs, but we know that 4 ERMIs is probably enough if we have that broader panel in the qualification study. And then we typically recommend spiking both a drug product and an RLD batch where possible. Sometimes we spike all of the batches, but I think this is what we recommend as a minimum, just in case we see anything unusual with the drug product. Do we see any boosting when we above the CL075 alone when it's in the DP? If we have the same RLD samples alongside, we can work out whether that's anything to do with the API or whether it's anything to do with impurities. So that gives us a kind of extra level of interpretation. And then we actually use, as mentioned, I'm not going to try and explain equivalence testing. I think people can talk to the biostatistician and talk to Sofie for that, because I have no idea how it works. But we've developed a method just to, again, compare those DPs versus RLDs to see if they're equivalent. I can't show you any data, unfortunately, because most of the data we generate isn't ours. But we do have some example data where we just created those equivalence limits for 3 batches of RLD, tested at 1 in 10 in a 20-donor cohort. And you can see the blue bars are the equivalence ranges. So this is plus or minus 1.5 times the standard deviation across the whole data set. So they're the RLDs in blue, and then we've got plotted one of the RLD batches that has been spiked with either CL075 and MDP, and you can see if it falls outside this range, then they're non-equivalent. So you can see that we can pick up responses to CL075 in the RLD in all the cytokines apart from interferon in this particular case. And the assay was much less sensitive in this case with MDP. So this was a very low level of MDP. So technically we did just about detect an IL-8 non-equivalence to IL-8. So this is sort of what we're doing with the DPs and RLDs. And we would obviously hope that we're seeing the drug products falling within the criteria for the RLD ranges. And I'd just like to finish in a couple of minutes just with some points for discussion. So I think these are all things that will be discussed in the round table, or if we have any questions in the panel session as well. I touched on whether we can use different approaches to different peptides. Also, we'll be discussing tomorrow oligos as well. How do we approach those? Do we take a similar approach here? I think the main thing to know is whether we have phenol or anything like that within the formulation. That means whether, if we don't have it, maybe we can go to a lower dilution, maybe a 1 in 5 rather than a 1 in 10. Everyone's mentioned number of donors. I think we all have similar ideas. So I think we're all in kind of the right ballpark range. I think we're mainly going to be stuck with fresh for now until we generate enough data to suggest that the frozen are a viable alternative. And I think that's going to take some time. Also wanted to mention, linked to the frozen PBMC, I guess, is what's more important. I know we like to check viability, but ultimately it's the functionality, it's that functional cytokine readout that's most important. So can we accept a bit of a hit on the viability if they're also still highly sensitive to the sorts of controls that they should be? I think we'll touch on this in the working groups about the pros and cons of testing in a 1 in 10 dilution, selection of controls that we've touched on as well, statistical analysis. I think that's still the one thing that I think we're all doing slightly differently. Similar approach, but all doing it slightly differently. Do we need to align on that? So that'll be an interesting discussion. And I think from what we're starting to see now is when we've run large number of studies, what happens when we see a difference? Is one difference in one cytokine that's very small differences but non-equivalent? Is that automatically a problem? And I think I know I can see Daniela smiling. We've talked about this a lot. Yeah, we're not sure where those where it becomes a problem. Where is enough of a difference to be concerned about? And then where we do need to investigate further, where we do see big enough differences to be concerned about, what do we do next? Do we repeat to see that it's robust? Do we need to block certain pattern recognition receptors to work out what could be happening, or isolate different cell types? And I think I tend to agree with Andrew's point, I think, about testing the DS or the API. That comes with a number of caveats and challenges as well, that we don't have a reference. I think the endotoxin limits for lots of these products are a lot higher than we would normally have in these sorts of assays as well, that we found out fairly recently. And obviously when we're testing the DPs and the RLDs, they have phenol, so they naturally suppress the responses anyway. So there's certainly pros and cons to testing the drug substance. So certainly be happy to discuss whether that should be a viable way forward. And I think right on time, I should be pretty much done. Yeah, I just wanted to thank all the guys in the UK and US teams, and thank you for your attention. Thanks.
Thank you, Noel, for the thought-provoking talk. And next, I would like to welcome Seth Thacker, Dr. Seth, for discussing the fit-for-purpose assay to assess innate immune response modulating impurities.
I want to thank the organizers for the chance to be here, and also the great talks which were previously given, I think, which set a big groundwork for my talk. My talk's going to be a little different focused, as I'm coming from what we would appreciate from an agency standpoint to facilitate the assessment from our view. So you've seen this before, you'll see it again, but this is our mandatory slide from our office. A quality product of any kind consistently meets the expectation of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It is what gives patients confidence in their next dose of medicine. Also, the disclaimer, this presentation reflects my views and therefore should not be used in place of regulations, public FDA guidance, or specific discussions with the agency. This slide has also come up several times today. This is one of our most favorite guidances, but this is what is the framework behind most of the principles we're going to talk about. It has been pointed out, even though it specifically references the 5 peptides, it has been applied. These scientific principles have been applied when appropriate. So immunogenicity risk assessments include an assessment of the impurities that activate the innate immune response. And to adequately assess the immunogenicity risk related to the innate immune response, the cells need to be sensitive and respond to a broad range of pattern recognition receptors, such as TLRs, NLRs, CILRs, as well as others. An assessment of innate immune modulating impurities is currently expected for products referring to the ANDA guidance, peptide guidance, or in which the scientific principles of the guidance are applied. So you've actually seen this as well, and it's good to see this being consistently representative and people are, it is being consistently. So from our standpoint, when these tests are being performed, we would like to see data from 3 batches of drug product that's been made from 2 batches of drug substance, and that being compared to 3 batches of the RLD. And then just to make our life easier, we would prefer to have it in a table such as this, where it's clearly delineated your batch numbers, each of the drug products batch numbers, as well as their date of manufacturers, the drug substance lots used to manufacture them, as well as their manufacturer date, as well as age at time of testing. These batches should be tested at different ages and be derived from separate manufacturing campaigns. At different ages along the life cycle of the drug. And if there are trends towards extractables, leachables, and aggregation, additional testing at the end of shelf life may be requested. So to provide useful information and actionable data, these assays need to be fit for purpose. This is what we've heard a lot of today, is fit for purpose. And the purpose of these is to demonstrate that your assay can detect a difference at an appropriate sensitivity. And these are, and we've seen as well, these are the parameters, the important parameters, minimal parameters that we assess in order to be able to demonstrate, we need in order to be able to determine if your assay is fit for purpose. So viability of the cells, sensitivity, specificity, precision, system suitability controls, acceptance criteria, as well as a detailed assay description. And all of this data submitted provides the underlying demonstration that your assay is performing consistently and with adequate sensitivity and has the appropriate controls in place to ensure the assay's consistent performance. So there are many of these assays that can be used. Primarily the focus up to this point in the session has been talking about PBMCs and whole blood. There are other options available, and there are continuing to be developed. We as an agency are agnostic to the selection of what platform you choose. We ask that you justify the selection in the readout, and that as you're choosing your platform, you need to incorporate the immunogenicity risk of your product, and that should be informing the selection. But yeah, so this is, the table just provides a list of options that are available. Some of these are obviously very new, skin biopsy models as well as organ on a chip. We don't want to stifle innovation, so we are not precluding anything. You just need to demonstrate that it's fit for purpose, meaning it has breadth and sensitivity of the assay that is commensurate with the risk of the product. So viability is a key component of these assays. This is coming from data that's been previously published. I'm just demonstrating that excipient components, in this case polysorbates or polyxmer 88, the number at the top indicates concentrations that's found in products and tested in these cell-based assays. You can see at levels that are normally found in these products, you can have a significant amount of impairment in viability. And then at the bottom, this is reiterating what was seen for what's been demonstrated by the other speakers. You can see definitely there's the APC populations appear to be much more sensitive to drug products or excipient components, which means they need to be, and this is why we are asking that these specific populations be monitored, because we found initially if you look at total bulk viability, you'll miss these smaller cell populations that are key responders in the innate immune response. We also ask, it seems trivial, but we also ask as you submit your data, provide a positive control in your assay demonstrating that your viability assay can in fact capture cell death. And once you've demonstrated that your selected drug concentrations that you've selected have a minimal impact on viability through developmental data, you can actually at that point, at the end of your pivotal studies, cell viability can either be directly confirmed at the end of study, or perhaps the viability can be inferred using established acceptance suitability standards, going back to what Noel was suggesting, that maybe as long as you can demonstrate sensitivity, an actual formal demonstration of viability may not be required. So as mentioned as well, we're talking about broad coverage. Because these assays are meant to detect innate immune activating impurities of unknown origin, and we don't exactly know what we're looking for, we need broad coverage. And as Noel had nicely in his talk, these range from cell surface, endosomal, cytoplasmic. And then when you're in your developmental data, when you're demonstrating that your assay has broad coverage, this needs to be done using a wide range to demonstrate that you can have coverage of a wide selection of these receptors. So the sensitivity should allow for the detection of trace levels of impurities if present. Determine the assay sensitivity using agonists for the different pathways, and importantly, establish what that sensitivity is in the presence of your drug product. And here's kind of an example of what you may see and what. So this would be, This is an example of what sensitivity may look like spiked in your drug product and media alone. But from an agency standpoint, we are interested in your assay being able to detect trace levels of impurities in your drug product. So when you report sensitivity of your assay, it's perfectly acceptable to report it in media spiked. But what we're truly interested in is sensitivity of your assay to be able to detect an impurity in your drug product. So simply stated, if you have to dilute your drug product, if you're having to use a 10-fold dilution, your assay sensitivity is not 100 picograms of LPS, it's actually 1 nanogram of LPS. So if you're having to dilute your drug product 50 times, you can see your true assay sensitivity to detect a drug product goes up quite a bit. This can be a problem with some drug products due to formulation impacts. We understand that. An option is, as been mentioned before, you can try to test your drug substance in a buffer that doesn't impact, or if there's a point in your manufacturing process before the formulation component that has an impact is added, you can try to test prior to that. We just suggest you leverage your manufacturing knowledge to determine whether there are samples prior to formulation that could be used to support the assessment without interference from the excipient. I was happy to see everyone. So in order to understand if the assay is consistent, we need to see that you're reporting the intra and intra-assay variability. And it looks like from our previous presenters, all of them were showing that quite well-controlled data. So going back to system suitability controls, so these are in place to demonstrate consistent sensitivity to low levels of a variety of model innate immune agonists, demonstrating the assay can respond to impurities in the drug product that may be present. These should be included on each plate for each donor. However, once the testing system is demonstrated to be fit for purpose through developmental data, you may only need a subset. So instead of using all the TLRs, you may, as previous talk Noel was showing, you may be able to select a surface, an endosomal, and a cytoplasmic to be included on every single plate. We just ask that you justify that selection in your submission. Another important thing that is sometimes missing in submissions is predefined acceptance criteria. So we ask that there is established acceptance criteria for your suitability controls to demonstrate that your assay performs consistently prior to the investigation of your pivotal sample testing. And this should be based on your developmental studies. Here are some examples of, this is not all encompassing, but this is some examples of what suitability acceptance criteria may look like. Your positive controls have a percent CV, so variability below 20%. Viability at your end of study is greater than 90%. That your low, medium, and high positive controls show dose dependency. And that greater than 75% of the positive controls for each donor are positive. And that your positive control is at least positive in 80% of donors. And these are examples and not exclusive. So it has been mentioned before, as you noticed in each of the presentations, readouts were all slightly different. There's many different readouts that are available. If you're looking at PBMCs, protein, cytokines, chemokines, you can look at the protein expression, the gene expression. Cell surface markers could be looked at, activation. People proposed using migration or antigen uptake. Similar to the platform, we don't necessarily have a predetermined preference for this readout. We just need to justify and demonstrate that your readout of choice can consistently demonstrate your assay can detect trace levels of impurities. So here is just an example of some of the readouts that are commonly seen, and many of these showed up in the previous talks. Here's kind of highlighting the different cytokines or chemokines and kind of how they fit into, whether it's an inflammatory response, anti-inflammatory response. We prefer that there are multiple readouts selected, which I think everyone is doing, so that they may inform on the activation of different potential innate pathways and innate immune pathways. We also find that readouts that are negative for all your suitability controls are not informative and can in fact hinder the interpretation. And once again, this table is for examples, it's not exhaustive, it was just some hypothetical biomarkers that could be chosen. To support a fit for purpose assay, we also ask that you submit a detailed SOP, which includes general assay parameters such as the cell source and processing method, cells per well, media, duration of assay, suitability controls used, plate layout, concentration of drug products tested, how the dilutions are being performed for your agonists and your drug products, and then how the viability was measured. And then there are specific ones depending on your platform. Examples of these may be if you're using primary cells, health status of the donors. The more information you can provide, the better. If you have age, ethnicity, that's all helpful. And then importantly, selection criteria for the recruitment of the donors, and then how long was the processing time? How long did it take in order for you to draw the blood till it was used for your experiment? And then cell lines have some other information as well, such as culture conditions, where the cell line derived from, what passages they were used on, et cetera. So for the analysis, we ask you provide the raw data as well as graphs with summary details. In that raw data, it's most helpful to provide a table that shows the linked individual measurements with cell recovery and viability, as well as the corresponding suitability controls for each donor or cell line run. We also ask that if you're using a standard curve in the analysis of your data, such as quantification of cytokine, you detail how that standard curve was generated and how the values and the data behind that standard curve were handled, and if any values were excluded. And then this is much simpler than what we heard about it. Yes, provide a clear, detailed statistical approach for how your drug product will be compared with the reference listed drug. And if you have multiple readouts, how you're going to incorporate those or how your analysis will be done. And just provide a clear, detailed clear details of how that statistical approach is being performed. And also it's important that you detail exactly how, if any of the outliers are removed. And with that, I'll conclude. I'd like to thank the Innate Immune Lab, Daniela, Verthelyi, as well as Mohan, my office main friend there, as well as other people at the FDA, Eric, Peng, and Cameron, Susan, and Steve. And that's all. Thank you.
Thank you, Seth, for the wonderful talk. And finally, we have Dr. Daniela Verthelyi to give us additional consideration for ERMI assays.
So hello again. I get to be the last speaker, which is kind of hard, but we'll try it anyway. You guys have seen that, so I'm going to go straight into the topic. And I thought, since I was the last one, that I would sort of reverse steps to the beginning and say, well, to assess the risk of a generic peptide, the whole idea is that we know what the patients are, we know what the way that the drug is being administered. None of that's going to be new for a generic peptide. So the majority of the risk is going to be regarding the API and potentially the impurities. And there is, to assess that the API is the same, there's really a number of assays that are being conducted, looking at the physical chemical properties, sequence structure, aggregation, impurities, and inactive ingredients. And if the risk of immunogenicity is not low, then there's a request to look at those impurities and how those impurities can impact on immunogenicity, right? And so if it is peptide-related impurities, as we discussed this morning, we would only be concerned if they're new or if they're in significantly higher level as the RLD. But when it comes to innate immune modulating impurities, we usually ask for them. And so why is that? So when we inoculate a product into tissue, right, there's a lot of immune cells that are embedded in the tissue. And grand part of the product is going to go straight into the draining lymph nodes. Some of it is going to retain within the tissue and it's going to make contact with those innate immune cells. And maybe there's going to be an immune response, maybe not. Maybe there's going to be some antibodies produced. When there are impurities with that product that are co-administered, then all of a sudden you get inflammation, a larger number of immune cells going into the area where the product is delivered, and a much higher possibility of that antigen-loaded, antigen-presenting cells are going to go into the lymph node, and they're going to express co-stimulatory molecules. You're going to have an optimized ability to present antigen, and they're going to generate an immune response. And with that, the possibility of now all of a sudden presenting new epitopes. And that possibility is maximized when we also have aggregates, because aggregates can also be taken up more easily by dendritic cells and can also stimulate B cells in an independent manner. So all of that's going to add to the risk. And the risk of antibodies and with it the risk of having changes to PK, reduced efficacy, and if we're really unlucky, a deficiency syndrome. So the concern, again, is that the impurities can change the quantity and the quality of the immune response. And because of that, as all of the previous presenters said, it's important that we understand what kind of assays you are producing and provide a detailed description of the samples and the acceptance criteria, detailed description of the assay, data demonstrating that the assay delivers scientifically sound and reproducible results with regard to viability, sensitivity, matrix interference, et cetera, and that you provide, and we ask also that you provide tabular data linking the response of the product batches to stability controls for each sample. And I'll get to that in a little bit. So they've all told you this, and I'm here to repeat it, but then I thought, what can I do as a last speaker? I figured that instead of going through it again, I would concentrate on some frequently asked questions. And these are questions that come up again and again, not only from sponsors, but internally. What I have here in the bottom, it says, dubito ergo cogito ergo sum, which means I doubt, therefore I think, therefore I am. We apply that at the agency, and we're constantly being challenged. And you heard one of those challenges this morning. It's like, do we really need this? What degree do we need it? That's a constant thing that we do at the agency, trying to really understand whether we're asking for the right things. So let's get to it. So some of the frequently asked questions. Are the detectable levels of impurities clinically relevant? That's one that comes up very often. And ideally, we would think that we would have an ERMI assay that would be able to detect and predict whether there's going to be a local or systemic innate immune activation, and really predict whether there would be NAbs. And more importantly, if we're really, really good at it, it would predict whether those NAbs would have a clinical consequence. We are not there yet, but we're getting close. So because we're not there, we combed the literature, we really looked at what we would know about, what concentration of potential impurities could lead to an immune response. There's a lot of data relating to vaccines. There's a lot of data for cancer, using these as adjuvants. But there's really not a lot of data, almost no data, telling you what's the lowest amount that can be a problem. So because of that, a few years ago, we did we did a study in primates that is what we called the limbo study. How low can you go? Basically, we administered PRR agonists to monkeys in lower and lower amounts to understand at what level we could detect an in vivo response. And non-human primates and us, we have a pretty good homology in terms of receptors, so we thought it was a good model. So basically, we did this scheme of giving lower and lower amounts of different PRR agonists. We took skin, and to make a very long story short, we came up with what we think are or what we were able to show were the lowest amounts of individual PRR ligands or TLR ligands that would induce an immune response in vivo. And we use this to sort of guide our understanding. Now, each of these different TLR agonists induced different patterns of cytokines, and we'll go back to that in a bit. But the patterns that we see where we've been able to compare them with human samples, those genes that we see in the NHP and the non-human primates and the humans pretty much correspond. So that gives us some confidence that what we're seeing is relevant. So with this, we established that trace levels of PRR agonists induce a detectable innate immune response in the macaques, but it didn't tell us whether it induced the antibodies. So we went back and we said, well, what level of stimulation could correspond to increased antibody production? And I'm showing you the example for endotoxin, where alone, as little as 10 picograms of endotoxin inoculated led to a detectable level in situ of IL-8. And you will see that the in situ effect is actually maybe more relevant than the systemic. And those levels really correlated with an increase in the titers or in the number of monkeys developing antibodies to a co-administered protein. So again, the idea is, yes, we see a local innate immune activation, and that one was associated with an increase in titer. To the extent that we can do, 5 monkeys per group, so not super solid, but some indication. We repeated a similar study more recently, and this one is not published, where we gave very low levels of endotoxin, 100 picograms, alone or together with a mAb, and the mAb was either as a monomer or aggregated, and when you had the aggregated mAb together with LPS, then you saw an increase in the amount of, in the number of monkeys that induced had detectable antibodies, and those were sustained antibodies, and more importantly, they correlated with a reduction in the levels of the drug in vivo, so they were neutralizing antibodies for all intents and purposes. So trace levels of impurities that bind PRR can elicit a local innate immune response, and that response can increase the risk of inducing ADE and NAB. So, you know, if we think about the assays that we have, and this is just a scheme of one of the assays that Miriam Mendoza and my group kindly drew for me, you know, it is actually very hard to think, well, this is the in vivo amounts that we see induce a detectable response in the subcutaneous tissue. This is what we can detect in vitro. It is not, it's compare apples to pears. We're completely aware of this. But on the other hand, I think that it puts us in a relevant ballpark. This tells us that where we are capturing with this innate immune response modulating impurity assays is the type of level of stimulants that we might see in a product that might induce an increase in an immune response. So another question that we are very frequently asked is, you know, well, yeah, we do all this characterization, do we still need this additional data to really understand? And that makes sense. I mean, if you think about it, if you look at all these individual impurities that I said, well, these are going to induce an innate immune response locally in the skin of a monkey. You know, you have things like, I don't know, 5 micrograms of CpGs or 1 microgram of flagellin. You should be able to maybe detect those with these assays, right? The thing is that impurities are going to be complex. It's not that you can have one type of impurity or one type of cell. There's many different sources of impurities, where there are microbial components, peptide synthesis byproducts, exogenous elements, aggregates. There's really a complex milieu of cells that are going to respond to those and interact and maybe have an immune response. So in a way, what we're doing is looking for assays that have an integrated signaling for the inflammation, for an inflammation or innate immune response. And B, we're usually using innate immune cells sort of as a canary in the mine, right? Because they're loaded with PRR receptors. So macrophage and dendritic cells, because they have the most PRR, they can be useful as sentinels, and that's how they're using them. And what we're measuring with these assays is not the response to LPS or the response to flagellin or the response to FSL. What we're measuring is what kind of overall impact are we making, or is the product making on the innate immune system? And is that going to be sufficient? And there's some product from Seth, some data from Seth who just spoke, that he, you know, in this study, he put concentrations of individual TLR agonists that were by themselves below the threshold. But when you start putting them in combination, all of a sudden you get a signal. Again, this idea that we're not looking, and this is why we're not looking for assays that can detect necessarily endotoxin, or can detect necessarily flagellin. We're looking for broad spectrum assays that can detect impurities or a combination of impurities that change the innate immune system. And, going back to the question, is this really necessary? And I thought I would share, this is actually a composite from a couple of things that we've seen, but try to make it as an example. And this is, your usual very nice characterization of a product, and you can see, you have 3 lots that you're comparing with another 3 lots, and everything's passed and everything looks gorgeous. But then when they go back and do the RMI assay, all of a sudden you see that there are some differences, right? In this case, it was a manufacturing change. You can see like the old lots are in blue and they're fairly consistent. The new lots, all of a sudden you have a signal and some disparities. So the RMI assay can detect impurities that are not in evidence using another analytical technique. And also, analytical techniques can discover impurities and not know what to do with them, right? You have a peak, it's a minor peak, and you don't know what that means from a biological standpoint. So that means that makes the RMI assay a very useful tool for us. So another thing that was discussed in the previous talks, but I thought I would touch on, is are cell lines sufficient to look at RMI assays? And back in 2015, we published an assay. We compared the use of 3 cell lines, raw blues, MM6, and THPs with PBMCs as a way of measuring RMIs. And in doing that, what we said was, none of these individual cell lines is particularly complete in the way of looking at impurities. Together, they might come close. And then there's this study from Wen et al. That actually came out of Lilly. And they did a lovely study really comparing the response. They have a DC internalization index assay, and they compare the internalization of antigen from primary monocytes versus THP1s. And you can see that things correlate pretty well. But when you start looking in detail, what you really see is that whether they correlate or not, or what signal they see or not, really depends on what the product is. So if you're looking at LPS, everything works, right? You stimulate your cells with LPS, everything works. But when you start looking at individual proteins, all of a sudden things don't work so well. So for example, if for bococizumab here at the end, the MDCs, which is the monocyte-derived dendritic cells, they would detect the presence of impurities in bococizumab by inducing IL-6, but not by inducing IL-1. So if you're looking at IL-1, you would miss those impurities. And THPs detected some, but not others. And it becomes a little bit more complicated. So this is where we are with respect to cell lines. Cell lines are high throughput, but they have a narrower scope and less sensitivity than primary cells. They also have a limited capacity to really assess for synergies. It's one type of cell line. You don't have different types of cell lines interaction. And they're really lousy at detecting aggregates. So for all those reasons, we don't love them as a platform. However, they are useful in assay development. They are used to complement PBMC assays, and they're very useful, we think, to qualify suitability standards and avoid what was mentioned this morning, the issue of drift. So there's certainly applications for cell-based assays. So one of the things that we've been mentioning is the idea of these multiple biomarkers, which may be useful because sometimes you would detect one marker, sometimes you would detect another, but they're hard to understand and hard to interpret. And this is basically the idea. If you have an RMI and you're only looking at IL-6, you're ignoring all these other activities, right? IL-6 is only present here, but all these other activities are being ignored. The idea is that because we're looking for impurities that we don't know what they could be, the idea is to monitor the different aspects of the innate immune system so as to understand whether any of those are being disrupted. So to give you an example, there are some disadvantages also. So Lantus versus Semglee. You all know Lantus is an insulin and a meat. Semglee is a biosimilar. It's interchangeable. It was deemed to be, and this was actually shown with clinical studies. They're reported, and they showed similar immunogenicity rate as Lantus. However, when we took 20 donors and we looked at the response in terms of IL-6 and IL-1, we saw that Semglee seemed to have a higher response. That can happen. You're only testing 20 donors. When we looked at all a combination of about 90 different markers, which we do, then we saw that it was really very few cytokines that had even a hint of an increase in response to Semglee than to Lantus. What does that mean? What does it mean? Does it really indicate that there's a difference? So the way, I think that this actually has to be looked at in a case-by-case situation, but the change in one biomarker may indicate that you have a problem with impurities. A change in multiple biomarkers, particularly if they are in one path, suggests that there really is one. Right? This is another example. This is from some old studies that we did with interferon beta, looking at them in M6 cells. And what you can see is that there's no differences in IL-1 alpha or beta, or 1 alpha or interferon gamma. But there is differences, particularly in chemokine expression and interferon-inducible genes. And so had we just been looking at IL-8 and IL-1, we would have seen no difference. But indeed, there was difference. In this case, I should say that this was a biosimilar candidate. It was a collaboration with Eduardo Mufarege, and it was his product. And the biosimilar candidate was cleaner than the original, so they were pretty happy with the results. But you can see how, by looking at a complete response, you have a better idea of what's going on. But then when you have multiple lots and you have multiple responses, you can have something like this, where, for the most part, they look the same. But then if you look in detail, there's, this one lot that looks maybe a little bit different than the other lots. Is that difference real? And it was mentioned before that one of the ways of looking at this is really integrating the data from all of those cytokines and all of those donors. And we have a number of ways in which we do that. And this is why we asked for the table with the actual data to look at, because we do integrate your data. And in this case, we did PCA. And one of the things that you see here is that the products, the different donor, the data points really group by donor and not by product, indicating that the impact of the donor is much stronger than the impact of the lot. And that sort of, again, gives us some peace of mind that we weren't really seeing a signal that was of concern. It was different when we did the same PCA with that data that I showed you before, right? So if you do that with this data, then you see that these are the old lots or the reference lots, and then they had one lot, the one here in orange, that was totally in a totally different space, and the two other new lots were also in a slightly different space. Now, when you looked in detail, they had lower inflammation markers, lower innate immune markers, so we wouldn't be concerned. But still, we need to understand what the general gist of it is. So, statistical analysis of complex data. Multiple readouts may provide a better assessment of the overall activation in innate immune paths, but can also mask or dilute a signal. So if you're going to choose multiple readouts for the innate immune response, you need to think about what paths you're monitoring and justify what readouts are being selected. Because just adding genes that are not going to tell us anything is just going to dilute the signal, and that's not going to be useful. Now, we do not recommend what statistical strategy to take and to analyze your data, but it is important that you develop this strategy to analyze your data, justify your selections, and consider what biomarker levels and number of responding cells may be useful, and so forth. With that, I want to thank you all for your attention, and I have one parting message, which is that the absence of evidence is not the same as evidence of absence, and to use in vitro assay data to inform immunogenicity risk, assays have to be fit for purpose, the results have to be understood with immunogenicity within the immunogenicity framework, and the potential for clinical impact of any differences needs to be considered. And with that, I want to just thank my partners in crime, Seth and Mohan, and the rest of the crew, including Eric and Camber. Thank you. We have break.
Thank you, Daniela, for a wonderful talk. All right, we have everyone situated, and that was a great session. So let's get the questions going. I know we have a lot of questions in the audience. I can see already Amy has. Go ahead.
Thank you. So the question is, for synthetic peptides, if you don't have bacterial or fungal contamination, what IIRMIs could be possibly in the product that might trigger innate responses?
Daniela, do you want to take that? Because your talk is pretty much relevant to what Amy was asking.
So there's two ways of asking that. And one of them is to think, well, looking at your process and thinking where can impurities come in, right? Can they come in from some of my equipment? Can they come in from some of my raw materials? Can they come in from my container closure? There's a number of places where impurities can come in. And you have to remember that PRRs are not just microbial. I know that we tend to think about them as pattern recognitions from microbial components, but they recognize a lot of alarmins. They recognize they can be, and even though we use TLR agonists as suitability controls for our innate immune assays, the idea is that we're looking for any impurity, microbial, non-microbial, that can change the innate milieu. So, these are products that are not going to go into patients before they see their commercial use. This is sort of, is there any hint that there could be impurities there that would increase the rectogenicity, the immunogenicity of the product? And that's the criteria that we use.
So aggregates might?
Absolutely. Aggregates, and there are a number of inert particles that, as you know, can cause the activation of innate immune cells as well. And there's a number of residual buffers and enzymes that can do so as well. So the spectrum of potential impurities is pretty large, and we don't want to limit it. We can't do it because it is, depends on the manufacturing process. Anybody disagree?
Not to disagree, but just to kind of echo it, the purpose, in my mind anyways, is not necessarily to show that, there's no microbial DNA in my product. There shouldn't be. It's synthetic peptide. If you've cleaned your equipment properly, of course it's not there. The purpose is to demonstrate that your product isn't any more immunogenic from the innate standpoint than what the reference product is. just using those suitability controls to really show and demonstrate that this particular pathway can be activated and I can detect it.
And if I may add one more thing, that makes it, I mean, we're very aware that that makes it extra hard. Demonstrating an absence is a lot harder than demonstrating presence of anything, right? And that means that your suitability standards are key and critical. And I want to sort of point to one thing that Noel mentioned, and that is the lot-to-lot variability, the source-to-source variability that we see. And, you know, the FDA is trying to work with us. We have an agreement with this. We're trying to develop reference standards that people can use to benchmark their assays. We think that that would really help all the sponsors to at least understand where their assays are. Again, everybody has a slightly different assay. And we talk about the IRMI assays. Every single assay that we see is a little bit different, right? So having good standards that are controlled, and you know, I mentioned, you need to look at have cell-based assays to make sure that there's no drift, that you really understand your assay. Demonstrating a negative, or at least negative to a certain level, is critical.
Mike here again. I see, I think, I doubt. Okay.
It's the other way around.
No,
okay.
that's my motto. Okay, I doubt. Okay, so these impurities at a certain level, again, which I claimed this morning are arbitrary, so 0.1 to 0.5, trigger this MHC kind of analysis, and it triggers the IMRIs, right? I think, well,
Not
anyway,
really.
my
No.
take on it is that if it has impurities, that triggers the testing for innate immunity or T-cell testing, all right?
No.
Okay, it seemed to me in the guidelines when I read it that it sort of is the trip for that. And part of it is that the peptides, it's unlikely the peptide has any like innate signaling ability. I think you would probably agree on that. You're looking for other things that are in these products. And I'm following up on Amy's question was, you know, I can't think of anything in this synthetic process really that would generate some kind of innate signaling. Okay, it's tested for microbial contamination. There's no LPS in it. Most of these other products have no LPS in them. A lot of your data was LPS added to products and testing for these things. But I think LPS is kind of, you can eliminate that source, right? So the problem is I can't figure out why we're triggered on these impurities to go and do this extra testing. And it's unlikely that the product is going to do anything. And if it comes up positive, we don't have a clue what that is or what that means, because there's no way to figure out what the signal would be. And to follow on that, basically, kind of question is to ask the companies that got up and talked about, your assays look great and you got great controls. How many positives have you come up with in all your clients, you know, that convinced you that they were contaminated by some innate, you know, pathogenic source, or of you guys, there's a 1, 2, 3, 4, 5, you know, are you run over with these innate signaling, you know, products that you got great assays, you know, put it on the table, you know, is it something you're worried about? Is it something that, you know, that you see all the time, or is it just really infrequent and it's not something we should be concerned about?
Just to clarify, the RMI assay is not linked to whether you have peptide-related impurities. And just so that there's no confusion, okay? Yeah, there's two independent assessments. Please, let's not confuse things. Yes, there are contaminants that can be in the peptides that can lead to innate immune activation. And as I said before, the aim of this is to make sure that they're not in the product. But they're not in the product, that's what I'm asking. Yes, we have. The short answer is yes, we have.
the same thing? Yes.
Yes.
Yes, yeah. Yeah, and then I think we all touched on this, is what do we do next? You know, what is a genuine risk to a patient? And I think that's the hard bit next. Right? Yeah,
I think that, you know, in the paper we published in 2015, or, you know, 2017, you know, we showed a couple of strategies to investigate and try to pursue what impurities might be there when you get a signal. But I believe that is one of the questions in our discussion group. So hopefully you'll join that and discuss it.
Sure.
I think we will have the
Yeah.
next question.
All right. So the question would be, so the IRMI impurities are spiked into our experimental material that we are testing to develop like more of a threshold, right? Now, is there a way for us to have a relevant peptide-based impurity? And I want to bring in the aspect of an intrinsic peptide-based immunogenic potential plus something that the peptide is associated with. So let's say we take something from the same manufacturing of that peptide from upstream, where you may have an associated impurity, which is related to that specific peptide. Could we use that as your relevant positive impurity control rather than the Hermes? Because I feel that if we spike something into a peptide or protein, we are making a control which will work in the assay, and it may also tell us what is the threshold, but it may not be the right impurity. So, and we did that with protein-based biologics. We started collecting upstream processed material, which we knew had more HCPs in it, or we knew it was prone to aggregation or misfolding, and we used that in the assay to say, okay, we are seeing something here, we keep on cleaning it up, and we stop seeing. And now when we do the lot-to-lot control specification strategy, we always ask, like, are we getting closer to our dirty upstream, or are we still close to what we thought was clean? So is that possible with the peptides?
From my perspective, I would say I think that that's feasible as an assay control. I'm not sure that it would necessarily, in my mind, replace the suitability controls where you're using known IRMIs. My reasoning for that would be is that I would expect that part of the reason that you're using those IRMIs is to demonstrate certain pathways that you know can be activated by your assay, and unless you have that characterized in that impurity that you mentioned, I'm not sure how you demonstrate for sure that, like, oh, I know this is a TLR4 agonist, and therefore I put it in, and now I see that the assay works. You see what I'm saying? So yes, it would be more related, and I think that would be a great supplemental control, but I'm not sure I would say it would replace all of the suitability controls.
I also think it would be a little hard. So the manufacture of synthetic peptides, and I acknowledge I've never made one, so bear with me, is a little different from a recombinant peptide or protein in that most of the time our sponsors are getting the API from commercially. And a lot of those times, that API is not sterile. So you're starting from an API that's not sterile and that comes from a different source. And that's yet another reason why it's so important to test for impurities. And that would make it, I think, a little harder for those sponsors to get material from upstream. And upstream is a little harder because then you have to deprotect your molecules. It's a little bit different from the recombinant process. We have, and Joey back there is doing some lovely studies, sort of trying to understand with the recombinant one, what level could you really test for IMI, or are there other components from the fermentation that are going to mask the presence of impurities and things like that. But I think that for recombinant peptides or proteins, it's a little bit of a different manufacturing train that's not the same for synthetic.
Hi. One of the things I wanted to ask is if there's any way that these positive results that you guys are getting in the IRMI assays could be linked to some sort of parameters in the specifications, for example. The product specifications are controlling for endotoxins, bacterial components, bacterial presence, aggregates, particulates. So what could we think about putting into the specifications that could inform of this potential risk? And also, innate immune responses sometimes occur without any more consequences, right? So when we get a positive result, how do we interpret this in the framework of clinical relevance down the line?
Okay, so when I started my talk, one of the things that I said is that the FDA continuously questions itself. I think you're seeing an example of that here. So we go back and forth, question, and there's a reason for that. We don't want to overburden the companies. So, but I also think that what I showed in my presentation is that you can do all of your API characterization, and we have had examples where sponsors have gone through the whole list and everything checks out, and they do the RMI assay, and there is an impurity there that they didn't see. We've also seen where there's no necessarily a signal until they actually spike the peptide with something, and then they see some synergy. There's some kind of impurity that they hadn't identified. All of a sudden, you see a signal. They're noting. So these are things that have happened that we've seen, and this is why we asked for it.
Yeah, but I was thinking more on how we can go a step further and figure out what's next. Now we have data that's informing on how many lots are we seeing positive. Can we pair up these with specifications that are on the high range? And if not, what are we looking at? What does it mean clinically? Because what I'm trying to say is that I think it would, with the amount of data that we have, perhaps we can go a step further. And like I was suggesting, trying to find a parameter in the specifications that help us understanding what these innate immune responses mean, which I agree that might play an adjuvant role. And if we don't put it in the patient, we're not going to see it. But I think it's important also to understand in what context these positive signals we are analyzing them in, right?
Yeah, so I think so it's not just in things that are actually activating the innate as well. There's other things within the formulation, right? I think I mentioned phenol a lot because it keeps me up at night. But essentially, that obviously suppresses, because we have to test it in 1 in 10, that's suppressing the assay. So are we seeing subtle differences between DPs and RLDs due to different amounts of phenol or different grades of phenol? So that's another concern that we have in particular, what is due to genuine activation and what's due to just less suppression. So I think that's another component. And I think we've touched on endotoxin as well. I think the endotoxin specifications for lots of these peptides is actually quite high if you look at the actual values. But you don't really see that because of the phenol suppression, again, with lots of these. So once you remove that, do you then see activation? And we see it when we test APIs all the time. They're much more challenging. So yeah, I think you're right around the specifications. I think you need to look at literally everything. And even if you think that's not going to activate the innate immune response, there could be something like taking the brakes off the suppression that leads to those differences as well. So I think, yeah, that's certainly something that we've been concerned about.
So I think also in relation to the comparison to the RLD, obviously that's a key thing. But you also have to remember that the numbers of donors that we're looking at in these assays are small. And at the end of the day, it's not about predicting what's going to happen in the clinic. It's about understanding the relative risk. So if you're going to have a very rare event that's going to be happening in a clinical situation that sometimes wouldn't even be picked up in a clinical trial when you're looking at thousands of people, you're sure as anything not going to be able to see that in 30 people. So there's an element of understanding what these assays can do and what they can't do. But obviously we're doing the direct comparison to the RLD, and it's really just about, as Daniela mentioned earlier, looking at a whole panel of different ERMs, a whole panel of different cytokines, so that we can see if any of these pathways are disrupted, and then just do that comparison and analysis compared to the RLD.
I see the value, but I'm missing how we apply that. I'm missing.
I think we're learning.
No, that's
you're right.
That I acknowledge it, and that's why I brought this question.
So I think that each company then has to, if they have a signal, they have to go back and look into their own manufacturing process where that signal is coming from. It would be very hard to give you sort of an overall response.
All right, we'll take another question here.
Sure. So first of all, it was a wonderful post-lunch session. I would like to thank each of the panel members. And my question is for Daniel. Your second last slide about the suddenly one batch showing up very high IRMI response as compared to the other batches. And then you did some investigation and found out that it was because of a different donor population. Okay. Now, this is something which you could find out, but for companies who are outsourcing their testing, would never be able to find out because the donor populations keep changing from time to time. You cannot have the same donors being used for PBMCs. Is there not something that calls for a kind of a regulation where PBMCs coming from a certain population of donors should stay put if we have to compare between batches? Because if this variability is going to be played out, for example, I'll just quote another example. If we have to assess ADCC activity for a monoclonal antibody, you cannot be generally taking PBMCs or NK cells from any donor. You have to first do the screening of the donor before you test them for ADCC. Something to that analogy, I would say, why can't we apply it for screening of donors while we look at inter-batch variability of IRMI response?
So, a couple things. First, that one lot out of 6 that was a little different. It was only a little bit different. It didn't quite reach statistical significance. Had it been really statistically different, our response might have been different. But we did see slight changes for one lot. Now you have to remember that all of these lots were tested for the same donors, all of them. So what you're saying so in essence, what you're seeing is a little bit of a different response from some donors to this one lot, right? So if you're taking 20 donors and you're comparing the response to 6 lots and all 20 donors, each lot is compared to the same donor to itself, basically. And I think that's, so having a stable of donors, I don't know if that would help you. I think that some of the companies actually qualify donors in terms of their ability to respond, to sort of weed out hyper-responders, I imagine, and hyper-responders, because there are some people that hyper-respond, and you sort of want to be in the middle. There's advantages and disadvantages. It's a tall order, I think, for companies to have a stable of donors that they can regularly get, that they've been vetted. And quite frankly, even if you vet them, you don't know if last week they were exposed to their kid who brought something from kindergarten, right? I mean, things change. The donors are not the donors. And the only way that you could do it repeatedly is if you froze a large amount of cells from the same donor. And that's one of the, strategies some people have used. There's advantages and disadvantages. As Noel was saying, you gain maybe in consistency, but you may lose in sensitivity, particularly when the freezing doesn't match well with the formulation of the product. So and I have to figure out what is the best strategy for your product and go for it. But sorry, one more thing. But really, not every change and the problem. Absolutely. And by all means, if you see a change and you don't think it's a problem, explain to us why not. We will look at your data and we will look at your reasoning behind it. Sorry, go ahead.
Okay, my question is related to FDA's recommendation to carry out immunogenicity testing near release and near end of shelf life samples. So now we carry out all these tests at the highest limit of impurity limits of specification or 100% of API concentration or against the RLD, all different in vitro tests. So what is the relevance of end of shelf life samples? Because of this recommendation, we can never get a first cycle approval of any peptide product. So I believe when we are carrying out at the near of release samples, these tests, they take care of everything. So would like to understand the significance of end of shelf life samples.
That's for me.
Means FDA panelists, means they can answer. I'm sure this answer should come from FDA.
Seth would like to have an.
Sure. I can try to answer it. So we actually, to be honest, this has changed a little bit. The end of shelf life may or may not be required. If you have data demonstrating through mid shelf life of your product showing no impact on IRMIs, ERMIs, and then there's no other data showing there's any increase in leachables, extractables, aggregation, you may not need data at the end of, for the ERMI assay to show data at the end of shelf life.
Okay, but in our deficiency letter or in the past presentation, it is always mentioned to use the 3 different lots of test products and RLD of different age group and especially near end of shelf life samples.
I think you can always provide justification sometimes. And we can say, you don't observe anything different. So you don't think it's going to be you can provide that justification.
Okay. So also there's a difference, I don't know the wording exactly that was sent to you, but there's a difference between process and product-related impurities. So product-related impurities would need to be assessed at the end of shelf life. These process-related impurities may not be needed.
Yeah, so process-related impurity will not increase, and the product-related impurities we have already analyzed at the highest concentration, so it should not impact anything.
Without saying, I don't exactly know based on not seeing the data specifically related to this, but it depends upon, yes, if there's any changes in process-related or product-related impurities would trigger maybe an assessment of IRMI at the end of shelf life.
Maybe if you can take this question back to your other colleagues and maybe rethink about this requirement, because what is happening if because of some reason, if we have to change the manufacturing site, we have, carried out this immunogenicity on the end of shelf life from one site. Now, because of some business reason, we have to change the manufacturing site. So is it still required to carry out now the immunogenicity end of shelf life from this new site? So those becomes very challenging for any ANDA holder.
We can share the data in our correspondence, right? We can look at it, right?
Yes.
Sure. Fine. Thank you.
Hi, I have one question for Daniela. You mentioned before, sorry, very nicely that the aim of this INATE study is to demonstrate the lack of any immunogenicity and compared to the reference. But my question is, for some products, actually, the INATE study is not really required, such as vasopressin, for example, if you remember that table. And my question is, I understand why, for example, adaptive studies might not be required because it's such a short peptide, it might not even be presented or whatever. But for the INATE, because there could be still leachables arising over shelf life or something and causing some problems. So why in this case, for example, the innate is not required? Thank you.
So yeah, I mean, vasopressin, I think, took a few headaches and a lot of discussions to go back and forth. I think that in the end, what was decided is that given the way in which it's administered, the clinical situation in which it is administered and the frequency, the concern about immunogenicity was low enough that we could forego that. And, you know, we don't ask for all these assessments for every product. You heard today that, no, we don't for orals. We try to, you know, avoid it for peptides where we think that there's not going to be a reason. So in one of my slides, it said, you know, if there's a reason to think that impurities could change the immunogenicity profile, that's when we ask for it. We didn't think that it would for vasopressin.
Yes.
Yes, Laura.
Hi. It's just, I think it's a follow-up on Monse's question, because I think as a consultant, what I'm seeing is just sometimes developers are struggling to see what is the scope of this testing. You're aiming to use this testing for a new drug application where you're trying to assess if you're having impurities that could potentially impact clinical outcomes. Are you using this test or determination of these impurities to demonstrate sameness of an appropriated new drug application? Or are you trying to build a strategy for evaluating clinical outcomes, immunogenicity outcomes? And I'm bringing this up because I think depending on what is your use for this assay, you're going to be validating it differently and to different set of parameters. So I think that would be useful to define whether or not you want to do this testing as part of your control strategy, quality control strategy, and therefore help you define acceptance criteria limits to ensure quality or sameness or clinical outcomes. I think this is something.
Two important things. Most of you have seen the latest labeling guidance for immunogenicity for proteins. And from it, you probably understand that the FDA does not consider the development of antibodies a clinical risk or a clinical consequence of concern. It's not an adverse event to develop antibodies. We're completely in agreement about this. The issue is that with generic peptides, and we are not asking for these assays for new molecular entities that are going to go into clinical trials, right? It's just in comparison with the RLD for products that are not going to be in clinical trials, because generics contemplate clinical trials, to ensure that there are no impurities in there that are going to lead to an increase in inflammation and immune activation that could increase the risk of an enhanced or different immune response. I can't hear you, I'm sorry.
It may not be necessarily linked to the generation of anti-drug antibodies, but you...
Explain more.
Yeah, you may have hypersensitivity reactions that
Yes.
are adverse events that could also be considered as immunogenicity, but not adaptive immune responses. And those, in that case, if you have a clinical concern over this innate activation of innate responses, I'm going to go away. You can manage them clinically, perhaps, and you don't need to do a clinical monitoring. So I'm stepping into the clinical relevance of activating these innate immune responses. And what would be the use of testing or predicting activation of these mediators in the clinic? Not only magnifying adaptive immune responses, meaning magnifying ADA responses, but per se, what is the relevance?
That's a loaded question. There's, depends on the route, depends on a lot of other factors. But yeah, definitely mast cells, eosinophils, They have PRR receptors, they have other innate immune receptors. Could you trigger something like that? Yes. Could you trigger reactogenicity? Yes. Again, we're trying to see, this is a sort of a threshold type of assay saying, you know, do we see anything that would let us to think that there's an impurity here that would be of clinical concern? And yes, that sponsors show that there isn't, so that we can, you know, they can go ahead and market the product with less concern that there would be a clinical event.
Okay, we're up on time. Just going to allow one more question. Go ahead.
So, all these innate studies, we came to know once that draft guidance for generic peptides came, and those are related to the process-related impurities. So, does it also equally applicable for other synthetic peptides, where the RLD is also synthetic? And if it is a process-related impurities, then why it is not applicable for other small molecules? What is the why specific for peptides these innate studies are required? Because when we talk about process-related impurities, that may be similar for other small molecule products also. And second, to the extension to his question, 2 stages, initial and at the end of shelf life. So if we do at the end of shelf life, do we really need to do at the release also? Because end of shelf life will be the worst case, then can't be avoided for the initial. Because we don't get both the samples together, in that it will run in 2 different assays.
So most of the times you're going to see a comment that says that we need 3 lots, and preferably we would like it from different manufacturing campaigns. And the idea is to make sure that you can do this consistently, that whatever products you make consistently don't trigger a response. For process-related impurities, we do not insist on end of shelf life. That's where we are right now. With respect to why for synthetic peptides that are that are generics to recombinant ones, I think that that we evolve, we learn and evolve, and like I think Mohan said this morning, sometimes it is not the best of rationale, but we're learning and we're evolving, and we're trying to use the best science to make sure that the products that get out there are safe and effective. And I can't necessarily tell you if you had a synthetic to a synthetic, would there be no risk? I think that a lot of times we are starting to ask for those also. But the idea, again, is to get the best science so that we can get the products that are safe and effective out to the public.
Okay, thank you. I would like to thank everyone, all the speakers. And then Cameron, would you like to give the closing? Thank you.
Good afternoon, everyone. So I'm Cameron Smith, and I've been charged with giving the summary of today's proceedings in light of our virtual audience will shortly be leaving us. So I think the meeting that we've attended today has been fantastic from my point of view. It's really been the first time that we've had a meeting of this scale where we've discussed this topic. We've brought members of both FDA and industry together to discuss this, and it's been really fruitful so far. So, looking forward to continued discussions tomorrow on this topic. So in the discussion today, we focus primarily on the challenges associated with assay design for both adaptive and innate immune response assays. In terms of the adaptive challenges, the validation of the assays at the in silico, MHC, HLA binding, and T-cell functional levels was discussed. In terms of In silico modeling, we looked at HLA binding to peptide alleles, the T cell receptor binding to peptide once it's bound to the HLA, and then there was a discussion of the need for in silico, in vivo correlation to validate this type of approach. There was some discussion on the limitations of these in silico assays in terms of non-natural amino acids or side chains, and then there was some concern from the agency about access to the algorithms for these approaches. We also looked at MHC binding assays, MAPS and T-cell response assays, and there was some discussion around the need for harmonization of the protocols for these assays. It was important, there was also a discussion around how it was important to, in the synthesis of the test peptides, that they be synthesized in an appropriate level of purity. The choice of cell lines, for example, PBMCs is a common choice, and also the viability of those cell lines must be established. In terms of the number of donors, the diversity of those donors, for example, healthy versus patient, was discussed. And then the selection of controls for these assays. So aspects such as peptide length, purity, propensity for aggregation, and T-cell epitope content were discussed. Then we also discussed defining the sensitivity and selectivity of these assays. And then there was some extensive discussion around the concentration of the peptides that are present in these assays. And then finally, for the adaptive, there was some discussion about the acceptability of the agency's current impurity thresholds. So then in terms of the innate immune response assays, some challenges that were apparent there were for the cell line assays, the choice of cell line was important, along with the readout. So for example, raw blue or THP1 blue cells can be used. And also there was some discussion of the cytokine assays, where we saw a discussion around the use of primary PBMC cells or whole blood cells. Whole blood in itself. Again, the number of donors was important. This could be based on interdonor variability, and it's usually healthy donors. Cell viability was discussed, especially in the context of formulation, impact on assay sensitivity. So for example, do you have the appropriate dilution or the concentration? And then should the drug substance be tested as a way of establishing whether you have interference from an excipient such as phenol, for example. We looked at validation of assay sensitivity to IRMIs that may be present, the choice of positive, negative, and suitability controls, which was critical to demonstrate sensitivity, and then also to ensure broad coverage of the innate immune receptors. And then finally, justification of the choice of the statistical approach to data analysis was looked at. So I'd just like to finish by thanking everyone who's been in the room, who's been involved in the discussions today, in the sense that these discussions have been in depth, respectful, and I think valuable, especially from the point of view of the agency. So I'd like to close by asking everyone to give a round of applause to all of our speakers and panelists that are present today.